Everyone. We're going to go ahead and get started. I hope you had a restful night. Thank you again for joining us for the second day of our ADEPT 8 meeting on drug dosing in pediatric patients with renal impairment. And thank you again to those who are joining us online as well. My name is Lily Mulugita. I'm the Associate Director for Policy and Research in the Division of Pediatrics and Maternal Health. We're going to start with session 3 today, which is future directions, and with the first session focusing on the role of modeling and simulation. We also want to welcome our colleagues from the European Medicines Agency. We have Colin Peter Jonas as well as Ephemios Mano online joining us, and they will be participating in the first session on the role of modeling and simulation. So without any further ado, I'm going to invite the first speaker, Justin Erb from the Office of Clinical Pharmacology, to give the first presentation. Thank you.
Okay, how do we advance the slides? Okay, great. Thanks, Lily. I don't know that my bio is online, so I'm going to just give you a brief background because I think it's relevant to the context of what I'm talking about. I've been with the agency for about 15 years in the Division of Pharmacometrics, and I've, during that time, been a reviewer the whole time in the last couple of years as team lead, and I see pediatric applications come through. We talk about allometry, we talk about maturation, and a lot of the concepts that get presented here. And, you know, when this workshop first came up, the idea of it a couple of months ago, I was really excited about this because the last year we've been talking a lot about extrapolation from adults to kids. And the idea, okay, is renal impairment in adults, does that translate to the same thing that's happening in maturation in pediatrics? And what are we thinking about there? So today, I'm going to talk a little bit about concepts that we deal with from at the time of a drug approval. So we have a certain amount of data that might be present for an application, and usually that's already been, the glomerular filtration has already been estimated, and we're talking about drug clearance being estimated at that time. And all of this ties back to the dose. What's the best dose for the patient? But that characterization of clearance during that time is really what we're after. At some point this morning, I'm going to get this to work. Okay, so this is the same disclaimer slide that you've seen everybody else present. Basically, I'm not talking about policy today. These are my own opinions, and I have no financial conflicts of interest to disclose. So I can think of a lot of different questions that we deal with. I'm not going to try and address all of them today. The first and foremost that we're after is what is the correct dose for the patient that we're dealing with. The next 4 or 5 questions all pertain to estimation of the individual's clearance. On the last one, it's something that I won't be able to get into today, but there is reason to believe that renal impairment impacts some exposure-response relationships, and this is always something that we should pay attention to whenever possible. So this is how I look at clearance data in renal impairment on the right. Oftentimes, probably the vast majority of times, we're looking at cough, cough, gulp because that's what most of the pharmacometricians in our field were originally taught years ago. Recently, we've had more guidance that suggests that MDRD needs to be brought into this, and you know, for pediatrics, definitely the bedside or modified Schwartz equation. Now, on the upper left, I'm going to clearance for the individual is a function of the individual's It might be a function of the individual's weight, their glomerular filtration, an age effect. Maybe there's a hepatic component. Other covariates are going to play into this function. And that's going to vary from the different pharmacological products that we talk about from day to day. On the lower left are two equations. They're arbitrary equations. We see many different forms of how clearance is characterized from drug applications that come in. The first one, the clearance is for a drug that is entirely renally cleared, where you're using an estimated glomerular filtration rate that's been centered to a median value and then raised to a certain power. And it also has a between subject variability component. The equation below that is linear, but it has both a hepatic component and a renal. And the renal might look like the equation above. These are typical examples of what we might see. Now, the example on the right is a typical way that we might look at this data. And for this product, the important thing is the clearance is shown in the bottom right. And there are two terms. There's a lot of products in the left term. I think there are 4 different components to that left term, and that left term represents hepatic clearance. And the right term is a Cockcroft-Gault application for the renal elimination here. But the point is that with this type of structure of an equation, your plot above is going to have an intercept well above 0, and that intercept is going to be reflective of your hepatic elimination. We talk about clearance for drugs that are completely renally eliminated. Let's just look at Sotalol on the left. I don't know that we need to entertain the plot on the right here. And oftentimes these relationships are shown completely linearly, and sometimes with almost no intercept, because that's the complete removal of the drug coming from the kidney. So this is interesting for me because one of the questions that we deal with, we go back to this plot. Okay, we have the majority of our data at the time of application was from 60 mils per minute and above, and then less than that. There are a few points, and that's somewhat informative for the moderate degree of impairment. But what about those with severe? How do we calculate how to dose in those individuals? And is the labeling that's coming out, does it have a high unmet medical need? High enough that we want to say, okay, what dose should we give these individuals? And so that question of the dose and the unmet medical need can be quite high for some diseases, particularly even in rare diseases where it's near impossible to collect that type of information. And that same goes for pediatrics. But that relationship, if there's no intercept there or other components that are contributing to elimination, and I'm seeing more and more that this relationship is often linear or close to linear. The one on the right is actually a power function, but that includes both an allometric component as well as a serum creatinine. But let's just talk about the one on the left. If it's truly linear like this, and you don't have data at that low end, it may be reasonable for some situations that we can infer for drugs that are completely renally eliminated, what might be happening at the more severe impairment levels. So the FDA has a guidance for industry out, pharmacokinetics in patients with impaired renal function, study design, data analysis, and impact on dosing. And when we get applications come through, and oftentimes we'll get the question, well, we only have sparse data in these individuals. Can population PK help us identify the clearance for moderate impairment or severe? It really comes back to, well, what's the data like? And most of the times, can pop PK do this? Well, pop PK is only as good as the data that can help support it. I mean, it definitely has the advantage of leveraging off of different sources of information and bringing things together and helping leverage potentially, for an example would be sparse data in the smallest infants, but you have a good amount of structural information in slightly older kids. Well, you could potentially leverage on this structure model to help inform the kinetics in the very smallest ones with fewer numbers of points. So what's in this guidance is really recommendations that are going to help support that type of modeling analysis. And I think one thing that we heard a lot about yesterday is something that in my 15 years these equations that calculate eGFR, I've known of 3 predominantly, and that's been driven by my experience of what I see from industry: Cock-Gall, MDRD, and bedside Schwartz. And there are a whole host of other equations that you saw yesterday, and I'll come back to for a specific purpose later in this talk, that that can potentially help inform this in the context of pediatrics and for particular disease states. So I think I'm going to be close to my time. I've got a red light flashing, but I'm going to jump ahead. I think we're all familiar with some of these core concepts here, but from a population PK point of view, we can break this into 3 major components: an allometric relationship. I think Roden and Anderson and Holford have established that the postmenstrual age can be used for a number of different applications to describe maturation. A particularly good example are for the gadolinium-based contrast agents, which are completely renally eliminated. This equation helped guide approval in infants and neonates without collecting additional PK data. So that's a case to be familiar with. And the last component then represents any remaining GFR or organ component. And in some cases, for how this was developed, this estimation of these, for the estimated GFR, these are a number of equations that were put forward, and they were developed based on different health status. If you look here, you've got some that are developed on healthy databases and others that are developed on CKD. And the ones that are developed on CKD, I would have loved to known about this earlier in my career, but here we're talking about these databases have infants, neonates, some of them. Some start from the age of 1 year. Most of your maturation has occurred by closer to birth or full term. But these equations might be reflective of what's truly happening for CKD. I can't really comment for those rarer diseases or non-renal diseases that might cause acute kidney issues. But this was an analysis that was done by John Wong when he was here at the FDA, and I pulled to it because it's starting to talk about the advantages of these other equations, these creatinine-based equations specifically. I think one of the challenges we face with the cystatin is how often that's collected. But what's interesting is here is half of these equations were developed on databases that had CKD, and a few of them did not have CKD. On the y-axis, you have drug clearance, and on the x-axis, you have a different eGFR prediction. Gadobutril is nearly 100% eliminated renally, so your line is linear from almost the origin, and in most cases, a linear relationship works. But what's interesting is that across all these different methods, whether they were conditioned on a CKD population or not, they're also somewhat relevant for the healthy population that the gadobutril data came from. This also shows that you have multiple different methods that seem to be predicting equally well, and that probably in part comes down to the inherent variability in the data. So I'll move away from this with a few thoughts. Current approaches to labeling have leveraged off existing data, yet we are challenged with how to label for more severe populations. Waiting for data that gives us a quantitative certainty, yet the remaining population suffers from a lack of dosing recommendations in the meantime. And surveys, okay, I'm going to jump ahead. I just talked about the surveys by Wang. They give us confidence for specific molecules, but also where those methods were derived from. Whether it was a context of pediatric CKD, it definitely supports use in that disease state, but there still is a remaining question about the determination of clearance for populations that may arise from different disease states, that potentially more rare diseases. And interesting, I think that for some of these products that are completely renally eliminated, we may be able to infer or even extrapolate a little bit in the context of high unmet medical need for products that are completely renally eliminated. There's ongoing PBPK work, QSP work. I know some of that was talked about yesterday, and I would love to bring more of that to your attention, but I know that Dr. Karim Azer is going to be talking about QSP in the next talk, so I'll take it from there.
Thank you so much, Justin. Our next speaker is going to be Karim Azer from Rutgers University.
Right. Well, thank you. It's a pleasure to be here, and I look forward to our session and panel discussion later today. So if you're sitting here today, you probably have this small device sitting next to you, like a mini computer called the iPhone, and that's been in the making for 60 to 70 plus years, depending on when you start the clock. And throughout those 60, 70 years, there's been an explosion in advances in computing technology that has allowed many things to happen: the creation of the internet, social sciences and networks, advances in measurement technology, and more recently, on the heels of the 21st century with the Human Genome Project, the application to understanding of disease and the genetic basis of disease. So what we've seen more recently is now an inflection point in how systems biology and the use of systems biology is helping us understand mechanisms of disease, and systems pharmacology approaches are helping us to understand mechanism of action and applications in translational sciences and clinical development. And three pillars of these applications are the models and the methods themselves, which are used in these approaches, and of course the data, primarily clinical data and clinical samples, as well as preclinical models that allow us to be more efficient and targeted when we see clinical samples. And today we'll talk about how systems biology and pharmacology, how we can bring that to the problem that we're addressing today in terms of understanding clearance of drugs and estimation of renal function. So why are systems models important in this context? That's because the kidney function is integral to many health and disease outcomes across a broad spectrum of organs, including cardiovascular, metabolic, even neurological, and cancer. And it's involved in regulation of many aspects of energy, metabolism, amino acids, etc. And therefore, when the kidney fails, it's not going to fail in isolation, and you need a systems approach. And that is what I'm here to convince you of, is that a systems approach will help us understand the multiple facets of chronic kidney disease and failure. And primarily, those are driven by metabolic and, as we know, hypertension, cardiovascular failures, and these are well-studied diseases that we can utilize and extract data from to inform a model that we can use for evaluating kidney function and extrapolating kidney clearance from adults to pediatrics. So I'm going to talk about three problems today that systems biology models can be applied to. The first is pediatric extrapolation, so assessing similarity of disease and similarity of response or drug response. The second is estimation of renal clearance through a model that can capture the renal function with age. And the third is assessment of kidney function measures and how we use systems models for that. So the next couple slides in a couple minutes, I'll go through each one and provide an example. So let's start with extrapolation. Well, the first aspect of extrapolation is evaluating, starting from the target that the drug is going after, and utilizing that target as the central point from which pathways and biomarkers would be the scope of defining the model that will be used. And once you've identified that, it's utilizing the adult data that's available and whatever pediatric data is available to be able to fit the model and generate virtual patients that have characteristics that you would want to be able to simulate, going across the severity of disease, genotypes if that's available, biomarkers, endpoints, and then the prevalence so that you have a weighted virtual population that is realistic. So we built a model like that for a rare disease called acid-sphingomyelinase deficiency, which is caused by a genetic mutation in the ASMPD1 gene. And it's a multi-organ disease that affects the lung, liver, spleen. And we built a multi-scale model that incorporates the primary pathway that is impacted by this enzyme replacement therapy, which is in the sphingolipid metabolism pathway. And we were able to show that we can fit and capture the key characteristics of the drug response to biomarkers of interest like plasma ceramide and sphingolipids, et cetera, and be able to capture complex behavior of the impact of the drug on the disease through these biomarkers. And what that allowed us to do is then be able to generate virtual patients for both adults and pediatrics, and then assess the similarity of disease between adults and pediatrics in a quantitative manner through simulation and evaluating how similar is similar enough in terms of response, and taking into account many aspects of the disease here. So the second problem is how do we evaluate kidney function early on during the maturation process and use that to estimate renal clearance? Well, this paper has advanced data here from autopsy of 71 kidneys and showed the relationship between glomerular generations and gestational age, kidney weights, and body weights. And more importantly, it's also providing an assessment of what is the sources of variability for kidney function and maturation, namely the number of nephrons and the glomerular generations that are the range of glomerular filtrations at birth, as well as the timing of nephrogenesis and the impact of that cessation of nephrogenesis on the number of nephrons that each individual will end up with throughout their life, and the number of generations involved. So you can imagine a model that is able to capture these sources of variability to be able to explain how the differences along these different variables could be explained changes with age, as well as changes with age much later on during in the elderly population as you also start to lose some of that later in life. And then the third example, I know Justin mentioned there's many equations here, and I'm putting one out. Well, the ideal scenario is that you would have one variable, right, or protein that is secreted uniformly and constantly throughout the body into the blood and excreted in the kidney, and varies with kidney disease. Well, it is almost unrealistic to have a variable like that doesn't depend on anything else, because the body is in homeostasis. And homeostasis means that there's going to be multiple factors that push and pull on each other to achieve that homeostasis. So systems models allow us to capture that homeostasis and be able to understand what are the underlying sources of variability that we need to correct for. Let me use creatinine as an example. Well, creatinine is actually a byproduct of creatine, and creatine is involved in the phosphocreatine shuttle system. And we all know that this phosphocreatine shuttle system gets triggered, especially during movement and exercise, and is an essential component of muscle metabolism. So you can imagine that changes in this muscle metabolism or the activity level of the person is going to lead to eventually different creatine levels. The second thing I want to point out is that the kidney is a high metabolic organ. And to support this active transport that happens, there's complex underlying energy systems. Different parts of the nephron will use either glycolysis or oxidative phosphorylation. So there are mitochondrial respiration considerations that we have to take into account as the kidney fails. So because it's a high metabolic organ, when you have nephrons failing, there's more pressure on the remaining nephrons, which will lead to changes in the energy states of the cells in the kidney and in mitochondrial respiration and biogenesis. Well, there's a number of models as I searched the literature for kidney models that account for different aspects of kidney function. For example, cardiovascular function or regulation. And these can be useful as modules in an integrated platform to allow for us to be able to use this integrated platform for extrapolation, for assessing and understanding kidney function, and how other organs like muscle or brain or liver can impact kidney function, especially as the kidney fails. In this table, I have highlighted a number of data sources that we can talk about during the discussion. I'm not going to go through them here. Some of them are clinical, some of them are preclinical, and some are like kidney on a chip or in vitro systems that can be very useful in filling the gaps in terms of data. So to summarize, kidney function plays a central role in overall health and disease, and changes in that function affect the whole system. And systems biology and systems pharmacology are ideal tools that allow us to measure and understand system-level effects, especially, for example, in this case, kidney function and regulation, and can be useful for three problems: extrapolation, assessing renal clearance with age, and understanding the impact of different renal measures like creatinine for estimating GFR, and how the impact of different organs play into that. And that requires building this modeling platform and a significant investment in generating data to inform this model over time. And with that, I'm going to close and thank you for your attention.
Thank you to the two great talks this morning that have set us up for a good discussion, hopefully. If I can invite the panelists to join us in the front. Are we able to have our EMA colleagues on the screen? So as they walk up, we have two panelists from the FDA, Jason Moore and Hao Zhu. Saskia, who joined us yesterday. You can join. Or you can sit too if you have any questions. Do you have questions? I have so much. Okay. You can sit if you like, or you can join. Okay. Thank you. Are we able to have our EMA colleagues show up as well? Perfect. So we have Colin and Estimus joining us from the EMA. Thank you. So we'll jump right in to our discussion on modeling and simulation. So as we start, we'll ask our EMA colleagues to start us off by telling us a little bit about how they're addressing this question of pediatric renal impairment at the EMA and how they're viewing the role of modeling and simulation currently. So Estimus, if you could start us out.
Yes, and first of all, thank you for inviting us to this workshop. And I think you nailed down the problem, and I see I could go through the slides of the previous sessions, and it's the workshop is very well made because it addresses the issues from the start. They have to do with the biomarkers, the availability of data in children, and the way to estimate and to measure renal function. All of these issues are very relevant, and the way the workshop is made is exactly that brings down what are the problems and the issues with assessing renal impairment in children. So for EMA also, it's really a multidimensional problem. We also recognize the problem with the biomarkers and the fact that there are many different equations that were used, but there's not a perfect equation to estimate, for example, eGFR in different age groups and different disease states. So there's not an easy way of predicting or estimating renal impairment in children. And modeling, we recognize modeling is a well-suited methodology to fill in the gaps. What happens in practice, though, it's I think our understanding of renal impairment in children, it's far away from ideal. We usually have few children with renal impairment in clinical trials, and the relevant dose recommendation in our labeling SMPC rely heavily on other data modeling and simulation. So we see usually population PK performed on adult data, and renal impaired adults are included in these data sets. And what is usually submitted to us is we see that the clearance, creatinine clearance relationship, it's estimated in adults, and after it's extrapolated to children. So it's a very simplistic way of predicting renal impairment in children, but this is usually what is happening in reality. It has been accepted in our submissions, and sometimes it has also led to those recommendations for renal impairment in children. So by saying that, I really applaud this workshop because it puts down very important issues on how we assess renal impairment in children. It's a very good start, and I would like also to give the floor to Peter if he wants to say something in addition.
Thank you. No, obviously I echo what you have said, Efthymios. And I think this workshop and this panelist discussion is an ideal moment to discuss some of the more technical aspects as well that we see when applicants come and they want to convince us of deriving dosing recommendations in children. I don't know whether we have to jump into that immediately.
Sure, you can start us off just talking about sort of where you see the current role of modeling and simulation in informing dosing, either for labeling purposes in the absence of data, or maybe to inform study drugs.
Sure, so I can speak to the first. So when we use modeling and simulation to inform on the dosing in the absence of data in pediatrics, what we typically see is, as what Efthymios has also said, is that a company would try to get an understanding of how different degrees of renal failure are related to the drug clearance, and then would estimate that relationship, typically with very empirical functions. And then usually what they will do is use that function and do a sort of a normalization. So they would look at an estimate for kidney function like predicted creatinine clearance. they would normalize that for body surface area, and then use that equation with the parameters estimated from the adult data to inform the dosing in pediatric patients, where they then would typically use a different equation for predicting creatinine clearance, like, for example, the Schwartz equation. So I think from our perspective, what we would like to see for the future is a more mechanistic way of doing that, or let's say at least the use of equations and functions that are more mechanistically inspired than simple, for example, linear or piecewise linear equations that are fitted from often just a few data even in adults. So I think if this would be a first point of discussion, I think amongst the panel, whether we can see opportunities for improving this, and whether, you know, what opportunities are out there to get a more mechanistic inspired model for that would probably extrapolate better or more sensibly from adults to children.
Thank you for that. Liping, would you like to add something from an industry perspective in terms of where you see the current modeling to address this question? Thanks.
Sure, I'd love to. So I'm a pharmacometrician, and I'm also a pediatric drug developer. When question like what's the role of modeling simulation in dosing for pediatric patients with renal impairment, the first term come to my mind is model-informed drug development. I can't think of a better setup given the challenges, the complexities, and the available knowledge to use this term in the pediatric dosing for patients with renal impairment to deliver the MIDD's full premise. And for me, it's the logic is simple. We look at a model as a mathematical representation of our current knowledge. What is our knowledge? The knowledge is a highly summarized format of the data. So if we ask, you know, what's the role of a model in this drug development? If you ask what's the role of data, the question doesn't seem that complex, doesn't seem that big, and people would jump in and say, sure, you use your data. Model is no different in a sense. It's an avatar version of data. It's a highly summarized level of our current knowledge. Just like Chris has mentioned, like iPhone, we have over the decades this rich base of knowledge, and we summarize in a model, and therefore we should use it. When in the situation like we talked about yesterday, with the guardrail of certain assumptions, certain situations, when we say model can replace data, what we really meant is we have gathered enough data for us to make a decision that with the tolerable risk that to facilitate efficient drug development. And when there's additional complexity, we talked about, when it gets younger, the ontogeny, the impact on non-renal part, the protein binding, what about safety, what about metabolites, etc. That's all the reason of using more of modeling to complement the data. Justin showed that guidance document. The slides he had is that giving adequate data to answer those questions. Let's be realistic. With this complexity, with pediatric subjects with renal impairment, think about as same as rare disease. You would never have trial-specific data to answer all those questions. So making the model even more important. So yesterday, what we're really saying is that we need data in combination with modeling to answer those questions. So it goes two ways. The modeling used there, going from patient selection, hypothesis generation, data collection, results interpretation to make it a better one. Because we just not being able to using data alone, never, not in this question, not in this setup. Last comment I want to quickly make is that I was very, very glad and really want to shout out to Lynn too, so clearly yesterday, saying that, the data collection doesn't only mean, or the model building doesn't only mean pre-marketing. If model is a highly summarized level of data, then the model only gets better when there's more data fitting in. So the regulatory guidance, the possible pathway of encouraging more work, letting people use it, collect that would make it helpful. We also heard yesterday saying the guideline, how to use a drug with pediatric renal impairment has over 100 of them, but in the drug labeling, there's only low 15-ish has labeling language. That means with this guideline book, goes to the physician, goes to the community, it has been used. Where are those data? Have they been formed model? So those are I was very glad to see the agenda. Next one coming up is labeling. I think from labeling to post-marketing to collaborative from research academia, how we best collect model. So as a pharmacometrician, to me, if we are holding this ADEPT 9 on this topic, we would not ask what's the role of modeling simulation. We would dive into detail. How do we best do that? I think that's the question. And from this 8, if we can giving more clarity on what situations that you have gathered enough certainty to give a straightforward statement of how to use it in this specific situation in people with pediatric patients with renal impairment, that'd be a huge, huge successful outcome of this workshop. So sorry it's long, it's hard not to get passionate about this. This is what I dedicated my career to.
Thank you. Thank you for sharing that sort of the marrying of data and modeling. Maybe we can hear a little bit from clinicians. I don't know, Sonia or Saskia, if you want to jump in terms of how you're viewing modeling in this space and how it's informing drug dosing.
Sure, I can talk both as I don't consider myself a pharmacometrics expert. I am aware of it, but I am definitely a clinician. So from a pharmacometrics standpoint, I think we've been talking a lot about extrapolating from adult data, but I think that we can also think about extrapolating from data from drugs where we do have the data for across the spectrum of renal function, from impairment to augmented renal clearance, and even for those patients who are on dialysis modalities. So, for example, I'm specifically in the anti-infective space, and there are some data of certain anti-infectives on patients who are on CRT, CKRT, where in the model they have shown the effect of what the filter and the cartridge do, and that we can isolate that effect from a model from one drug and possibly actually apply it to a model for another drug if there's similar biophysical properties, targets, and then be able to use these adapted models and then be able to validate in real-world data and then end up doing simulation to be able to come up with dosing regimens. I think from a clinician, the issue that we have, I know, Saskia, you're a critical care doctor, is at the 2 a.m, you have your very sick patient, and the question is, do they really behave what the model says? And I think that as I've been more in this space and also just listening to everyone yesterday, that as good as our EGFR equations are, I don't think that they will ever perfectly capture what's actually going on in real time, especially in a very dynamic patient, and even in our work in a quote patient with stable renal function, even when we are getting creatinine and cystatin C and using the U25 equations, it only explains 60% of the clearance of a drug that's supposedly primarily renally cleared, and even when we look at other covariates, maybe we're explaining 70%. And so I think the question that I keep thinking about, what are those drugs and what are those populations where we absolutely need to get the drug exposure right, and instead of maybe trying to perfect our way of measuring GFR, should we be investing in therapeutic drug monitoring? I'm in the model-informed precision dosing space. I am all about using observed drug concentrations and then using a population model that is decent, that is as close to that patient as possible, using that as the Bayesian prior and being able to actually model and individually model and simulate that patient's PK/PD profile in real time so that we can inform drug dosing for that patient. Because I think much as we like models and they do describe a lot of variability, you know, when you talk with critical care docs, they're going to say, but my patient is different. This is why we don't have, you know, sweeping guidelines in the PICU, because my patient has 5 other meds to consider and volume overload and whatnot.
Do you want to add to it?
Well, I can completely echo what Sonia just said. What I would like to add is that well, and I think this is also a call to those of you here, that I think we should come together and really make a plan how the existing data, as we showed yesterday, there's quite a number of POP-PK papers. There's a little bit of PBPK modeling done in this field. Leveraging the data, and just what Sonia said, there's quite a bit of data that we probably did not find with a review or don't pick out on antibiotics, on renally clear drugs, where people have actually looked at different stages of AKI. They probably may not call it AKI, but just say, well, we looked at creatinine or creatinine clearance, and then indeed try to see if we can figure out how far down can you go to extrapolate. And personally, I would prefer to have PBPK models and further develop and get this information in these models, so that you don't need a lot of new information and not a lot of new data to come up with dosing advices for patients. And what has been said just earlier today is the big risk of postponing collecting samples from patients in the post-marketing stage is that in between, indeed, after registration of the drug, there's no guidance. And I think if we can leverage these models a little bit better, make them better with existing data, we can, then the label could provide the dose, and then the post-marketing studies could be used to actually confirm that dose is correct, and then in an iterative stage. In the clinic, things that, I don't know how it is in Europe. Here, I think it's about similar. In Europe, we have big issues with the medical devices regulation. So any calculator that someone proposes is going to encounter a lot of hurdles to be accepted when it really relates to individual patient care. So we did have a calculator on our website for the pediatric formulary, very simple. Just you put in the age and the weight of the patient, and it would give a dose. We had to take it off because we couldn't afford to go through all these steps to get it, to get it approved and to be safe for use in clinical care. And that's something that needs to be taken in. It's really, say, make a calculator and use it in the clinic. It's not that simple anymore. And if companies want us to do this, then they need to provide us with that tool that can be incorporated in electronic health records of like the big companies that everyone is using.
Oh, thank you for sharing that perspective. Maybe we can combine a little bit of the next question, and if anyone in the audience has questions, feel free. But we've talked about sort of the current role of modeling and simulation and where it is, but can you maybe articulate what kind of challenges or gaps that exist that you think we can fill with that data you were talking about? Maybe we can start from our FDA colleagues. Oh, Saskia, you wanted to start? Hal or Jason?
Yeah, I'd be happy to continue that discussion here. I think those are great questions. To me, you know, I don't think the modeling and simulation techniques has any issues. The gaps probably exist from the knowledge of our mechanistic understanding for this underlying biological process in pediatric patients with renal impairment, and also the data we have. I think everybody has already talked about it. So we lack of, you know, some of the data we can use for model building and validation in patients, for example, in young age, and especially with some specific populations. I think those are the major gaps. I've seen this, and to me, you know, to collect sufficient information would be very valuable for us to fill in those gaps. And one question actually is, I think I wanted to come back to our panel, is I heard the discussion where, you know, we can sort of, you know, get some model prediction to come up with a dose recommendation and get into the post-marketing space and then fine-tune the model with additional data would be somehow a strategy that the community would like to take. Because initial stage where we give some instructions in the label, it's not the perfect model, but we know that it's better than just a physician gets up at 5 o'clock in the morning to figure out the dosing by themselves. Now we give some instructions, but we know it's not perfect one. There's some uncertainty associated with it. But we know that with more data coming along with the post-marketing experience, then we can fine-tune that model. Would be something that, as a community, you would consider as a possible way to sort of narrow the gap and actually provide some instruction for patients and physicians too.
Well, I think that could be a solution. At the same time, there have to be safeguards that it actually does happen. The risk that I foresee is that then the drug is put on the market and there won't be funding to collect these data. And then it comes onto academia to get funding to collect this data. That won't happen. So similar to like the oncology drugs where they put on the market, well, we even don't know if they work. There is the companies need to collect these data. I think that there could be a similar thing for these kind of data, because if you leave it to academia and to the public funders, I think that would be a little bit unfair. Right. And it won't happen.
Right. I'm just saying post-marketing could be, we request a certain post-marketing studies in order to ask the sponsor to track, to collect additional information so that we can use that information to fine-tune the model we have, and maybe we're going to change the dosing recommendation, let's say 2 years or 5 years down the road, but we can give some initial recommendation based on our best knowledge of by using modeling simulation. At this point, by the time we're going to move on to prove this product for use in pediatric indication. Would that be something, this time frame of 2 to 5 years would be something, do you feel to think that's a good strategy to move down the path, sort of narrow down the gap a little bit, or do you feel like there's still too much risk, or there's case by case, and if so, what type of criteria you would consider? I'm just going.
Ethmios has a question.
Yeah, Ethmios.
Thank you for bringing out this topic, because the other question I have to the panel is, I think we have to make a distinction between post-marketing and real-world data. And I don't know how much we can rely on real-world data to validate these models, because, for example, comparing different literature data, we see a lot of variation in standard operation procedures, in analytical methods. So do you think there's a way of using this real-world data to validate maturation functions and models that can be used after in the labeling? And if so, are there any specific measures we could put as regulators or as a community in place to do that?
Turn to answer.
Well, I'm not going to maybe try to answer, but I'd like to piggyback on that from maybe the industry side of that. Ethmios, I agree with you completely. The challenges that I see, and I think the questions that I have in terms of how to overcome this is how do we unify what we collect? And when we're looking at a post-marketing setting, a real-world setting, yes, as you mentioned, Saskia, we have insurance databases, but again, the data collected within those databases do vary. And quite frankly, when we look at pediatric data, it's sparse. It's not as robust as the adult data that we have. So I think the onus is on us. I will own that as well, along with everybody else. But, how do we harmonize what we collect? How do we make it so that we can use that, even if we're talking about specific pharmaceutical products, but also as class products? And how do we use that information and leverage that together? And I think, if you make it a post-marketing requirement, commitment, request, whichever one it is, I still think we need parameters around how to execute.
I recently had a chance to review a paper from Mark Smith-Davis for eBiomedicine about new blood pressure percentiles for children, and they used 8 large American hospitals and used the EMR. And I think we can get some more detail about the principles with a similar approach, and one would have to start with an ethics application about that. But For instance, I mean, you have to have something that you can measure, and the measured levels that are mandatory are for vancomycin and for aminoglycosides. So even just pulling large amount of data, and there are probably millions of vancomycin levels in the world, but nobody has put them together. So pooling all of this and then looking at perhaps using artificial intelligence to come up with models that go beyond our scope, because I think it has more, the volume of distribution, the fluid overload, all of those things need to be to really accurately look at that. But then we could maybe take those principles that, especially in the first 2 years of life, and then extrapolate to drugs that are having the same transporter mechanism, for instance.
I think in principle, I think that makes perfect sense. It comes down to the execution. And even when we look at pooling data, even vancomycin levels, were they all collected at the same time? Were they all collected the same way? Et cetera, it becomes, pooling data is theoretically great, having done it, and when we present this data for drug applications, it does become a challenge in terms of how do you interpret it, and we're really looking at apples to apples. So I agree with you in principle. I think it comes down to, again, how do we harmonize and ensure the quality of the data will give us the answers we need. And I think that's still, in my mind, a big hurdle.
Yeah, I want to go back for a second to the idea that was mentioned in the panel that we don't have the right equation yet for pediatric renal impairment. And just remind everybody that one of the last slides I showed was a table of equations from the Edith Mahari-Stark, and that there are equations there that describe glomerular filtration. And if you think about how we describe clearance in a pop PK model, we have the allometric component, ontogeny, but a lot of the Mahari-Stark the equations listed in that paper have a database that already includes the maturation in those equations as well. So I think there are probably ways of constructing from that database for the CKD population an equation that might be useful in moving forward. It's something that I didn't catch before this workshop, but looking at what's out there, I think there's some things that we can work with moving forward.
Sure, I just have one question. I think Justin mentioned earlier the difficulties with having sufficient data at every degree of renal impairment. So even in adults, we have insufficient data in the most severe. I was wondering how much data would we need in pediatric patients to validate these models? So if we had some observed data in mild to moderate, is that sufficient? Could we extrapolate from one population to another? I was wondering if you could comment about that as well. Maybe Jason?
Thank you. Perhaps I can start by addressing that question. Thank you very much for it. I think that it's very case specific. So referring back to the ceftazidime avibactam example, there was a very specific scenario where both drugs were very highly renally eliminated, and we had a lot of data in adult patients that went beyond normal and mild. So we had a pretty good understanding of the effect of renal impairment on pharmacology in adults. And then, as you mentioned in that example, we had some data in pediatric patients with normal kidney function and with mild renal impairment. And I think we had a couple patients in the moderate category as well. So there we felt pretty comfortable extrapolating beyond based on the available data. And that situation, because it goes back to what one of my previous panelists brought up earlier. We had a lot of data generally about the effect of renal impairment on eGFR, on clearance. And because it was so highly renally eliminated, we felt comfortable using that data to support what was going on for patients who would be administered this drug combination.
Do you want to take them now? Do you want to take them now? Or wait?
Okay. One question. Due to the likelihood of sparse sampling and or small sample size in pediatric trials, pop PK modeling for pediatric models are often stabilized with adult data. Can you comment on bias that inclusion of adult data might introduce? Additionally, can you comment on complications introduced by use of different kidney function calculations in pediatrics and adult clinical trials as it relates to the modeling data set? Bedside Schwartz versus Crockett-Gault, for example.
The panel?
Yeah, hi. So this is Peter from EMA. I think I can speak to the question on technical problems that arise when you use different prediction equations for adults versus pediatric patients. I think that usually complicates things in the setting where we typically first have adult data and then try to extrapolate down to children to do a dose selection prior to getting any information. Having different equations for predicting estimated creatinine clearance generates a sort of a discontinuum in that extrapolation that we're doing. So recently there have been new approaches to that, where, for example, O'Hanlon and Alford published an equation to scale GFR from premature neonates all the way up to adults. I think those kind of equations are more helpful in that context, because at least they take away that additional complexity that we introduce by having different prediction equations in adults versus children.
One thing I'd like to add to that as well. I mean, so I fully recognize that using different equations in adults and pediatric patients is going to create it's going to add some additional variability and potentially a little bit of confounding there. But I think one thing that's going to be important as well is going to be to understand what the impact of that variability would be, or the impact of potential overprediction or underprediction in one scenario may be. So I think there may be some cases where we have a pretty good understanding of the pharmacodynamics, perhaps, or where we have a fairly good window for safety, where we may be comfortable with a little bit of uncertainty if we feel that it fits within the range, within a window where we feel relatively confident about the effectiveness and safety.
I don't really have answers about the biases there, but one thing that I just wanted to bring up, so I'm also a complex care doctor, and I take care of a lot of young adults in my children's hospital because adult hospitals are afraid to take care of them because they don't know how to do weight-based dosing, and I do include them in my models because I think that they do still have quite a bit of physiology that is similar to similar-sized patients that are in the pediatric world. Whether that is the right answer, I don't know, but I think it's a population we need to think about as medicine advances and children with medical complexity becomes the predominant population that's being hospitalized because we're much better at our care outpatient. We need to think about when do we switch over to adult equations when their creatinines are 0.1, 0.2 because they have very little muscle mass. I just wanted to make a couple of comments on some of the questions and issues that came up earlier. I'm 100% a believer in real-world data and using that to validate models. I think where we come into issues about funding these kind of studies, whether from an academic standpoint, is people want to know, how does this affect outcomes? Like, does it matter that we're missing the exposure? Does it affect outcomes? This is such a problem in infectious diseases. We are fortunate that we don't have high mortality rates in pediatrics. And so what are the right endpoints to say, yes, it does matter that we are time over MIC, AUC over MIC. Even in the vancomycin world where we are getting lots of concentrations, So far, what we've kind of shown is that with AUC-guided dosing, we help with nephrotoxicity, and not everybody even believes that, but is it improving other clinical outcomes? And so until we're able to also show, like, yes, it matters that we're getting the exposure right because it affects outcomes, it's an uphill battle for us in academics to be able to show that. Well, the last thing I wanted to put a plug in, it was very briefly mentioned yesterday, but the Pediatric Trials Network, which is funded by NICHD under the BPCA Act through Duke, they do amazing work in scavenged opportunistic sampling. That's how I model my personal work after, and they have hundreds of sites where they are getting sparse data, but it's across 100 different sites, and so ultimately it becomes rich data where they are depositing their data in the DASH repository, where I do think there are data existing on drugs that we are using in renal impairment, and we just need the minds to look at that data and see how we can come up with drug dosing recommendations and models.
If I may add to answer the first part of that question coming from online, that is with the huge generally the huge imbalance and the different type of data that we collect from adults to pediatrics. I think technically that's renal impairment or not, it's the same issue we have when we look at pediatric data in general. From technical point of view, I see the data as prior knowledge. Like in the Bayesian analysis, we always try to assess the impact of prior on the data, on the question we're trying to analyze. So while pulling the data, using adult try to help to reduce the uncertainty, increase the understanding of pediatrics is great. Meantime, the sensitivity on the level of leverage we could have on adults should always be there. That's one. Treat the same way as you do Bayesian analysis, and assess different borrowing, how that impacts your outcome. That's a given. The second thing is that beyond this quantitative part, there's a lot of practical differences in the sampling frequency, food status, sampling time. We talked about diurnal differences yesterday. All those things can be different in the clinical data collection, and that shall be considered in the model building as well. And meantime, be aware of confounding that Jason mentioned, that when we look at renal function, some of the growth-induced change has already been there. There's another thing that we don't see that in the literature very much, but the other day I was studying a drug that was approved in the label. What intrigued me, was find very interesting, is that there's a, not for renal impairment, but there's a approved pediatric dose from young age all the way up to 18. And if I follow that milligram per kilogram correct dose regimen, let's say I derive the dose of 200, that's for the illustration. And then once the subjects get into 18, the adult approved dose is actually 100 milligrams. So I look at this as like, what happened? This kid gets older and he needs to take half of the dose. So when I walked back and went through tons of literature on how this compound, developed in the history, what happened actually is the little things. When we talk about proportional, yesterday we said, okay, you are cutting the adults by half if your weight is such. What adults are we talking about? So for this particular compound, the healthy subjects have a higher clearance. The sick patients usually happen in the 60s, 70 years old, a certain organ impairment, they have a higher clearance. So what happened is that the scaling down for this compound, I can only do diagnosis, I didn't do this, it's not our company's product, is that they take the scaling down from healthy, which has a higher clearance, they give this drug. So, and then they proportion to that. So, of course, by the time they get 18, they get a higher dose. But for the adults approved, it's 18 and all above, they have 100 milligrams. So this actually happened in the label. So talking about the technical details of modeling adults and peds, those factors we shall all consider, and that's not unique to patients with renal impairment, but, technical questions. So when we do model the assumptions, the detail, the communication with clinicians, all that shall be done better by our pharmacometricians.
Another question from the online. Would it be a possibility if you set up an open collaborative pediatric registry for collection of relevant data with acceptable parameters and variables that would serve drug labeling?
One of the things that NICHD has supported is the MPRINT initiative to collect a lot of this type of guidance as well as data. I mean, I think it's early on in the process for them, but they have that intention at least to do that. So, I mean, that may be something to ask Langview and Sarah Quinney at some point, but I think they're at least have the mission to do that, but I think it's a great idea. Others, any comments? Saskia.
Well, this doesn't provide us concentrations, drug concentrations. So if you also want to know what happens with the exposure of the drug, so as mentioned by Sonia, the PTN has a long-standing opportunistic sampling project with all these sites, GCP level collection. So if a company has a drug and has to do post-marketing studies, they could ask a network for those sites, if the drug is being prescribed, to collect the data that you want to have collected. So I think, but maybe I'm thinking too academic here and I'm missing some points. But I think that would be a infrastructure that could be used for this, because they know how to do these kind of like opportunistic sampling studies, so that you not only can collect the electronic health record data, which have a lot of issues indeed, and don't provide you the information, is my model right?
You know, while we're on the topic of data, one of the things, there's been a lot of calls for more sophisticated models, more mechanistic models, more artificial intelligence, and it would seem to be requiring different types of data. We talked about real-world data, but I was struck in Kareem's talk, he showed some autopsy data, which is kind of unique in the fact that the system pharmacology approach has different type of data that would inform those types of models that are not what we're used to collecting here. If all we do is collect more of the same type of data, I don't think we'll get enough in this regard. So maybe just to ask the panel, and Kareem, you especially, what are the gaps you see from a more mechanistic approach where we could kind of put our heads together and target filling in some of that information, particularly around model elements where assumptions are key and we're maybe a little fragile?
Yeah, thanks for that question. Is it on? Yeah. So I think part of it is there's data, and there's also mechanistic knowledge and understanding biophysical laws and processes that we can characterize into a model structure. I've talked a little bit about how we can modularize the different aspects of a systems model so that we can dissect different sources of data and integrate that into a greater. Now, the mechanistic piece, I think, for example, there's models of cardiovascular regulation that have been well characterized. I think in terms of gaps of data, there's several layers to this. There's data that's available that is not harmonized and we don't have access to, and there's already been some discussion on that and how do you consolidate it, to the last question. There's data that you can generate, but it's difficult to generate because of the patient aspect. And then there's data that you need innovations in measurement technology or different measurement systems to be able to fill these gaps. I think the last part is the most exciting, because now you're talking about things that you're not able to generate today that you need to generate. For example, we've seen sequencing technology that has evolved over time and has been had significant impact on our ability to understand, genetic basis of disease, under the underlying pathways, because these advances have significantly reduced the cost and the scalability in terms of how much you can do, and now we can do whole genome sequencing. in a very similar way, in assessing kidney function, there's preclinical models that are now being developed, like kidney on a chip, like organoid models that we can use to understand how data that's generated, like through this autopsy, how some of these parameters can impact filtration rate, and be able to characterize these relationships into a model that can be representative of clinical function. And then the third piece is, being able to innovate and invest in the measurement technology so that we can measure things today that we can't measure, or we can measure them in a way that is easier to implement in a patient setting.
You want to add something?
I just have an idea. Sorry, I'm speaking scratch, but when you look at the pyramids of CKD, right, you always have the broad base of CKD stage 1, and in both adults and in pediatrics, the top with CKD stage 4 and stage 5 is a very small number. So regression analysis, however you do them, will need points at the extremes in order to be robust. And with the current sampling, you will have very few data points in the bottom. So I'm just wondering what the panel would think about using the group of patients that we use the least when we are shying away from the neonates, but they have physiologically low GFR, and they have physiologically low recruitment of nephrons, and they actually change in the first 4 days quite considerably. What does the panel think if we adjust for ontogeny of the transporter proteins, if we adjust for the total body water changes, and so on, to actually use that group of patients who would be in the hospital and accessible to come up with the bottom extreme and then the normal healthy children, and then extrapolate from the regression lines with robust data points in both ends?
Anybody from the panel? I mean, I like this concept because you're really pushing the boundary conditions of some of those equations and, going where you can have a better data density collected. Anybody else?
I mean, I think you can also simulate that in different model systems to be able to supplement the paucity of, data in in end-stage renal disease, to be able to understand how, when you get to very low number of nephrons, how much does that place a burden on the system, on the kidney, and it's more than a relationship and bookending it. It's also understanding how the system is going to respond to change, because as I mentioned earlier, the kidney is a high metabolic organ, so you're putting stress on multiple levels. It's filtration, but it's the active transport, and it's the underlying energetic system that is trying to adapt to be able to do that. And with that, you're going to create, there's going to be regulatory mechanisms to, say, thicken the arterioles to be able to sustain more blood flow to smaller regions that are still trying to keep things going, and being able to take advantage of that understanding. Maybe if that will lead to a simple relationship, but I think starting with the understanding and building towards that is sort of more of a systems approach, and achieving what you're looking for.
Yeah, I'm just thinking, because from the adult world it becomes more clear that when you have renal failure, previously it was thought that if your GFR goes down, your renal secretion goes proportionally in the same way it goes down, but now we know that may not be the case, and it may be dependent on the transporters. When you extrapolate that to a 3-year-old with complete renal failure, and then you extrapolate that to a newborn who is relatively a preterm who is otherwise healthy, I'm a bit worried that this balance between change in GFR and the impact of the renal transporters may get us a little bit in trouble. But I would like, again, to make, again, a case for PBPK modeling, because I think any equation you make to use in POP-PK modeling, you still need a lot of data. So this bottom-up and top-down combination of data, I think will, in the end, if you can put this in, it can put this data in PBPK modeling and improve the models again and again and again. I think that may help us also to answer this kind of question. And we're doing this currently also in pregnancy, where we develop these models with placental perfusion, and we have limited data from just a couple of women, but the models are quite spot on when we validate these limited, like 8 women or 6 women and fetuses, without having to study like 50 women to get the same results when you only do POP-PK. So it's and of course, and we've also just put out a more statistical way on how to determine for how many patients do you need data for these PBPK models to actually be able to be certain about the confidence interval, instead of the, for those of you more into PBPK model, the 0.5 to 2 times. And it really helps you also to think about the number of patients that you need to include data for. But this mechanistic thing and the difference in mechanisms, I think is important to keep in mind. At the same time that, well, if there's a wide therapeutic window, why do we care? I mean, I think that's something that we also forget. Like, we try to tease, it has to be perfect, but does it really make a difference for this specific drug? I think that's, and often we've run into this when we did our own PPK modeling, and then in the end we said, well, the therapeutic window is so wide, it doesn't have to be perfect, and then we would backtrack. But I think that's something that you need to start off with. What are the risks, and what are the risks of underdosing, what are the risks of overdosing, and then titrate what kind of data you need and how precise you need to be. Use the Schwartz 1, 2, 3, 4 peers. Will it make a big difference, or will it not make a big difference?
Hal, you wanted to add something?
Just to quickly follow up on that. Again, I understand that the concept of using neonates, but the question I have is, like we just talked about, is, one condition is pretty healthy. You have the body stress level is pretty normal, but under renal impairment patients' status, that the body stress is totally different. Are we actually looking at the same issue or not? And also, I was thinking about the question actually, somebody raises online, just by looking at, again, the imbalanced sample size that is going to potentially bias the results from the estimate from the clearance. So I think that's a fair point. So that actually tells me that how important it is to look at trial design aspect. It's not just to say I can throw any data into the model to generate the results. We have to design the trial in order to make sure we capture that information. That's number one. Number two is what I wanted to remind you is that we don't have to generate the so-called statistical significance. Our focus is to look at the trend. As long as we're looking at different ranges of the entire span of clearance, then we can actually capture the trend properly. I think we probably are okay. So I think that should be a focus if we want to design it. And also going back to the question, if we're using the different equations that may generate a different dosing for pediatric patients under different subset of populations, I think it is fairly possible. I'm just going back to your point, is that you have to tolerate that tolerable. We have to look at uncertainty. Then we have to see if we can tolerate that uncertainty comes along with the dosing. One way, of course, we can do this is we can consider a range of doses in pediatric patients under different scenarios, and we can see if there's anything we can monitor clinically, not just the PK-wise. For example, efficacy or safety. If we have those signals, we can potentially titrate patients and make sure that we're end up in appropriate range for that individual patients based on their whatever parameters we plug in. So I think those could potentially be solutions to address some of the issues we're facing here.
Thank you. Estimates you wanted to add to that?
I would like to follow up on the PBPK and just to mention that we see the value of PBPK and also EMA regulatory system, but we still need data to qualify this model. So these models might be able to work with less data when they can be applied in the context of a specific drug development. But in order to reach this state, you need to be able to build the model and qualify the model on quite a few data coming from different molecules with different with similar physicochemical properties and also similar ADME properties. So I think it's a good solution, but it needs an investment in collecting this data and qualifying this model. And the same applies for QSP models. But I definitely see the value of these models and the potential to develop these models in the near future.
Lynn?
I can only agree.
Yes, I'm going to preface this question with a full agreement on what Hal and Saskia said just now, that it should be sort of scientifically justified, understood why we're collecting these data and to use them appropriately, not just to say, oh, here's a piece of data, let's throw it in. But having said that, my question to probably Hal and Jason is, what is the population of renal impairment in adults that's being used to assess? Because my understanding was it's a lot of older individuals who actually have renal impairment but don't necessarily have chronic kidney disease from a disease. So I was just curious, and again, I'm not advocating that we do something less well, but what is the population that's being studied on the adult side right now to support the dosage recommendations for renal impairment?
Take that question. Perhaps I can start us off. I think it depends. So right now I work a lot in oncology, and there we have fewer healthy participant studies, especially as it relates to renal impairment. And so a lot of these patients instead would have the disease of interest, and I think they would have more of the CKD, but it would also depend on their specific cancer as well. So I mean, certainly in something like renal cell carcinoma, you would expect a lot of the kidney disease to be from the cancer itself. But then perhaps in melanoma or a gastrointestinal cancer, you would not, and then they would have it for a different reason as well. So I think perhaps not a great answer to your question, but it does, I think, depend a little bit on the specific disease state.
If I may add on top of that, I think that's absolutely right. That's a great question, Lynn. I think, yes, you're absolutely right. To most of the indications we see, we're just getting patients with renal impairment. We're not really looking at specifically what causes this renal impairment. Having said that, for some of the indications, we know there's a high prevalence of, the disease may cause renal impairment. So there's an interaction or a combination of those. We do actually recommend that, to do the renal impairment study earlier and generate a dose recommendation and test that in the trials, so in that specific combination of disease plus renal impairment. So I think that could be, I think could be a supplement to information we can get from the phase 3 trials.
I know we will be closing very soon unless there is a question, but I think what I've heard most is that there is a real big potential to leverage data available, generate new data, and then that can really help move these models forward. I don't know if you wanted to add anything before we close, but this really goes into the next session after our break.
The only thing I think that struck me, I think Li Ping started us off by saying, MIDD is really endorsing this approach, and I think with MIDD, any sponsor knows that they need to landscape and really assess where data exists. So I was glad that Saskia brought up the PTN and other scavenged data sources. I think getting more comfort with having access to that information, using it in submission, getting feedback from our regulatory colleagues that it added value, but also addressing the need that we have multiple equations here that we're dealing with. That's now in 2023, but it's like This morning when Lynn and I were getting a coffee, she used cash to pay for it. So they're still accepting cash, which means that these equations are still okay.
Don't shame me.
Yeah. No, I'm just using you in a positive light, Lynn. But I think it just illustrates the fact that, we don't have to be satisfied with the current state of affairs, but that doesn't mean it's not useful and it's not serving the needs. And I think this whole session is really in the spirit of, can we do better? How can we do better? Collectively coming and looking at, generating additional data, improving different diversity of models, and then revisiting this at Lynn's next workshop.
And I do see our next speaker. So, okay, great. So we're going to resume with the next session, and our first speaker is going to be Jan Markard from BI. So if I can have Mark come up, please.
Okay. Oh, thank you. Yeah, thank you so much for inviting me. It's a super important workshop, and it's really a privilege to present an industry, but also pediatric clinical trialist perspective on this topic. I work for Boehringer Ingelheim, and the views and opinions expressed do not necessarily reflect the views or positions of Boehringer Ingelheim. I would like to start with pointing to this guidance, because guidance in general is very important for us as industry. The better the guidance, the better the clinical development plans, and the better the clinical trials. And it's great that we have this workshop, because I think we acknowledge that there is a lot of work to do from a guidance perspective when it comes to PK assessment in pediatric patients with renal impairment. And when we would think about guidance, I started just with a couple of very general questions that might be important and that we have to ask very early to ensure that we can really build in plans for how we address these questions in patients with pediatric patients with renal impairment into our clinical development plans. And here are a couple of those questions. So when do we need the data, and what kind of data do we actually need? Is the drug that we study likely to be used in pediatric patients with renal impairment? So to have a kind of also prioritization. Is impaired renal function likely to affect the PK of the drug or not? And do we have data, PK data from adult studies? Do we even have dedicated PK studies in adult patients with renal impairment? Because if we think about modeling extrapolation, this can really help us. And I feel it's really important to define exactly the PK data needed to plan evidence generation appropriately, and this early on, to ensure that if we need certain data points, that we can build this into our clinical development plans. And then it really comes to the point, do we need full PK data in a sufficient number of pediatric patients with renal impairment over all age ranges? Or do we have situations where PK modeling would be sufficient, and PK modeling could include POP-PK or PB-PK, and where we would use data from adult patients with and without renal impairment and data from pediatric patients with normal renal function? Are there situations where we could think about post-marketing data collection only, or where we would just monitor PK? We discussed very complex individual patients and cases, and the question is, might it be just pragmatic to ensure that we can measure the drug levels in time and appropriately to monitor and to ensure safety? And are there situations where maybe no data are needed at all? For example, for topical use only. Maybe this sounds trivial, but to have kind of very clear guidance on where and how we have to study might be a good starting point. I also have a couple of other considerations for clinical trials. Because what we really try to do and where we want to approve with clinical trials in pediatric patients is to bring innovative and breakthrough medications to pediatric patients as fast as possible. and to be very pragmatic with these kind of studies. So when we now try to bring in special populations, we also want to ensure that we do not affect the overall clinical development timelines for populations that might have a very huge unmet need for new medications. There's also a question if we think about including special populations, and this could be patients with renal impairment, are there also maybe for certain drugs other special populations that might be relevant and that we haven't really discussed at this workshop? This could include hepatic impairment, or it could include significant other comorbidities, especially these complex patients that we discussed and that we see in pediatrics might not only have challenges from renal impairment side, and how can we really ensure that we have safe and appropriate dosing recommendation for these patients. One other consideration is when we think about including patients with renal impairment into clinical studies, could this compromise the primary endpoint? Because whenever we design our studies and we deal with relatively small number of patients, it can be a challenge from a power perspective. And we follow more and more an ITT approach. So therefore, if we bring in kind of high-risk patients, especially if it's chronic treatment, if you want to assess an endpoint after 6 months or even later, and we include patients with renal impairment or significant comorbidities, what would be the chance that this patient could in the end contribute to the primary endpoint, or would they drop out? And therefore the question is, could it be an issue for the overall trial? And then it really comes to the question, what are alternative scenarios in case we really have to assess and get data prior to approval within a clinical study? Are there options maybe to have an ancillary study on a kind of open-label active PK assessment in pediatric patients, especially when we think about what do we need for modeling and simulation? Do we really have to include patients with renal impairment in a placebo-controlled study, or is what we really need the PK data in these patients? And once we can ensure that these patients are within the window, can we then assume that they would also have the same efficacy and safety that we see in pediatric patients with normal renal function? And then, of course, and this is what we also discussed, we might have to really challenge ourselves when we discuss the key in- and exclusion criteria when it comes to renal function. There is a kind of mechanism to include patients with lower EGFR, and for sure there has to be a very clear rationale to do this. I would like to provide just one example here, and this is one of our own studies with SGLT2 inhibitors, where we looked into patients with pediatric patients with type 2 diabetes. And in this study, we excluded patients with an EGFR of less than 60 based on the Sabitelli formula. And we had a good reason to do this, because we know based on the mode of action with SGLT2 inhibitors, and this is glucose excretion via the urine, we do not see a lot of efficacy in patients with diabetes and reduced kidney function from a glycemic control perspective. So these patients need a certain renal function. So therefore there was a good reason not to include them. However, there are also situations where we have to include them. And SGLT2 inhibitors are, again, another great example, because they are not only medications for patients with diabetes, they can also prevent progression of kidney disease. And this is why we will now do even a specific study in pediatric patients with kidney disease with SGLT2 inhibitors to study exactly this. And I think this shows that there is not really a one-size-fits-all approach, but we have to ask this question and define it early on and really need a very good rationale. It's also about the age ranges. We discussed this earlier, and also about the concomitant medications and other comorbidities. that we oftentimes exclude, but that are also very relevant when it comes to dosing. So there is a lot of work to do on this side. and this is just to summarize what could also help when it comes to guidance. And I prepared this presentation prior to this workshop, so if I would have to do the presentation again, it would be a little bit different because we had great discussions about a lot of these topics. Starting with the first topic, I feel that we had very good discussions and about a potential consensus about what are really the right equations that we should use, or could there be additional guidance for specific populations to ensure that the industry studies really pick the best equations and formulas in alignment with regulators, academia, and experts. Again, we have to be very open-minded and think about innovative trial designs and how we can include pediatric patients with renal impairment with the aim of keeping the studies feasible. Because again, with pediatric development, we have really unmet medical needs that we want to address, and most pediatric patients have a normal renal function. So we still want to improve there and still be faster and try everything we can do to be faster. We discussed, the potential of post-marketing, real-world data, PK collection, but also the point that this needs to be funded in the end. And this is a point where it would be challenging for industry to do it on their own. So whenever we do in our pediatric studies, post-marketing, or in general pediatric studies, whenever we can collaborate with academia, whenever we can collaborate with existing networks or registries, it really helps. And therefore, I would really like to go to my last point. And this is, I think, key to be successful. So there really needs to be great collaboration between regulators, pharmaceutical companies, academia, clinicians, and patients to address this topic. And therefore, I'm super happy that we have this workshop today as a starting point. And I hope that we continue the discussions and also look forward to the panel discussions to share additional thoughts. Thank you.
Thank you so much, Jan, for that excellent presentation. Our next speaker is Soo-Young Choi from the FDA Office of Clinical Pharmacology.
Good morning, everyone. It's my honor to start the last session of this workshop. The title of my presentation is Renal Dosing in Pediatric Patients and Labeling Considerations. This is the disclaimer that this view is my own and may not represent FDA policy. So during this workshop, we've delved into a range of different topics related to renal impairment in pediatrics, from the assessment of renal function and how to translate adult dosing to pediatrics. And altogether, I'd like to start my presentation by reiterating the ultimate goal here, which is to identify safe and effective dosing recommendations for pediatric patients with varying degrees of renal impairment, and by providing clear and actionable recommendations to healthcare providers. So this is the example that ceftazidime and avibactam that was brought up several times during this workshop. And adults with renal impairment, depending on the degree of renal impairment, the renal dosing is either dose reduction and or changes in frequency. And the goal is to identify the dosing regimen for pediatrics with renal impairment, and how do we get here, and what information do we need to get that answer for that question mark. That is the whole, ultimate goal of this discussion. So this survey result was also shared by different speakers during the workshop. So it's not surprising that the renal dosing for pediatrics is not readily available for many drugs. And based on this survey, only 15% of the drug that were available with specific recommendations for pediatric renal impairment in major pediatric dosing handbook, only 15% has some sort of recommendation in the label. So why is it so challenging about it? And we also discussed this a lot during the workshop. Then the main reason is simply that we don't have any data in this population, pediatric patient with renal impairment. And the first reason is obviously the rarity of the condition. However, the exclusion of patients with some or any degree of renal impairment, that's something that is discussed during this workshop. So, and for some cases, for some instances, it makes sense to exclude those patients. We have a reason to be cautious, and we have the reason to exclude these patients, pediatric patients with renal impairment. And some of the examples were well articulated by our industry colleague. But there are some other instances where patients were just excluded, no clear reasons, and this has been the issue for not only for pediatric patients with renal impairment, but also so many other specific populations. So here's an example of an antiviral drug, and this drug is renally eliminated, and that means the drug's concentration is higher in adult patients with renal impairment compared to adult patients with normal renal function. And based on all available data, for adult phase 3 trial, the enrollment criteria was creatinine clearance 50 or 60 ml per minute. And when we reviewed all the data, ultimately it is approved for adult with adult patient with the GFR minimum eGFR of 30 ml per minute per body 1.73 meters square above, meaning that this drug can be taken in adult patient with mild to moderate renal impairment. That was how it was approved. However, in pediatric trials, the enrollment criteria was much more stricter, that any degree of renal impairment was excluded and for no clear rationale in this case, and it ends up being there's no specific dosing recommendation in pediatric. So in this case, though, this might have been a missed opportunity to collect any data, meaning the safety, PK, and efficacy data in this small population. So I wanted to highlight that in some cases, it makes sense to exclude the patient, but not all cases. In some other instances, they're just automatically excluded. The next topic is that, so in the end, in most cases, we'll have to rely on adult data because it's not going to be possible to collect adequate safety, efficacy, PK data in children with renal impairment. So to be able to do that, to understand how adult renal dosing is constructed is important. So the first approach that we commonly see for adult renal dosing recommendation is I would describe as exposure matching, meaning that if you see twofold higher exposures in adults with renal impairment, like say severe renal impairment, as compared to those with normal renal function, you reduce the dose by half. So you kind of exposure match to people with normal renal function where we have plenty of data in terms of safety and efficacy. And the other approach that I honestly see more and more often, at least in antiviral areas, there's I named this threshold approach, that meaning that despite higher exposures, companies provide adequate safety database to support a standard dosing for subjects with renal impairment. So in this case, the recommendation is something like no dose adjustment is recommended in patients with severe renal impairment or even end-stage renal disease. And the rationale is that available safety data, either in this population or either in a setting of higher exposure, support the use of full dose despite higher drug exposure. So my point here is that to extrapolate anything from adult to pediatric, in terms of dosing in renal impairment patient, that understand how adult dosing is constructed is important. Then all the other points that we've discussed a lot during this workshop is translating other renal impairment PK based on what equation we use, BSA indexing and de-indexing. Those are all important points, but so first we need to understand how adult data recommendations are made. And so in the next couple of slides, I will be sharing some examples of the currently available labeling language. And this is not intended to choose the best one or arguing that this should be way to go. So every drug is different, but rather by sharing these examples during our discussion, I hope that we can think about the ultimate outcome of this discussion. Like, what do we want to do in the end? How do we want to inform healthcare professionals? So first approach this group of recommendation is outlining a general principle without specific dosing recommendation. And these three examples that are common here is that first, the label recognizes the lack of data, so no available data in pediatrics with renal impairment. However, it recognized that you need to look at the renal function in kids as well, because that's what happens when adults and drug is renally eliminated. How to do that? The first label suggests that just simply age-appropriate assessment without giving any specific equations. Then how do we do a dose reduction? The second example shows that it should parallel that recommendation for adults. For example, if you recommend half of the dose reduction for adults with severe renal impairment, you should do the same thing for children. So this is a set of examples currently available. And the next set of examples is that recommendation for adults are similar to recommendation for pediatric. So the first example, again, is the same thing, ceftazidime, avibactam, and the other two examples I have here, that the recommendation is the same for adults and pediatrics. Say the first one, for example, 50% reduction in adults with a creatinine clearance 30 to 50, and pediatrics, eGFR of 30 to 50 based on Schwartz. This should not be interpreted that we view as interchangeable. We've discussed a lot during this workshop. These two equations are different, and there's differences and divergence between the two equations. But the more important thing is that despite that differences and some limitations, if available data are able to support this type of conclusion, at least that this is an actionable recommendation, and using two commonly used equations to respective population that healthcare providers can rely on the information without going through all different calculations or without trying to guess what's the best way to for renal dosing in children. So if data are available, this can be an option. Like, again, this should not interpret as the equations are interchangeable, but available data support this type of recommendation. The last set of example is where the actually recommendation for adults is different from the recommendation for children. And I put these two examples in valganciclovir and dabigatran. In both cases, for valganciclovir case, adult dosing is based on creatinine clearance, and pediatrics is based on very unique formula that's 7 times BSA creatinine clearance. And for dabigatran, adult cutoff for full dosing is creatinine clearance 30, and pediatrics is eGFR 50. So there's numerically is different there compared to the labeling example number 2. Here, the point that I'm trying to make in this slide is actually that these recommendations reflect the study design and dosing used in the trial. So going back to the point, study design stage and enrollment, what is the best practice and how do we improve that, our current practice, given that in some cases we'll just have to stick to the the way the dosing and enrollment criteria that was placed in the trial design stage for making our recommendations. So, with this is a summary of my presentation, also summary of this workshop. That first, that we do not have a dosing available for pediatrics with renal impairment for many cases, and this is due to multiple challenging aspects, but also further discussion is needed to establish improved labeling recommendation, and this is why we are all here. And with this, I'd like to conclude my presentation. Thank you.
Thank you, So-Young, for that great presentation. So we're going to transition to the last panel discussion. I'll ask the moderators, Lin Yao and Bakri Al-Zarqa, to come up, as well as the panelists to come up to the front, please.
So maybe we can start the fourth and final panel discussion by taking clarifying questions for the last two presentations. Any questions online for the last session? From like this fourth session. So maybe I can start with a quick question. So we talked about, is there any key or situation or certain indication to choose between PK modeling and actually getting real data from pediatric patients from renal impairment? Is the medication going to be used more in specific CKD population, like an anemia drug or a bone disease drug? Does that make a difference in choosing if we're going to do modeling versus actually collecting real data?
Well, so I guess I would ask if you're asking, should we do a post-marketing requirement to actually get that data, since most of the time we can't get it pre-approval, versus modeling and simulation, or is it really a choice? Should we be doing both?
I mean, the question is is there certain things about the drug itself that may lead us to one of those two ways, or?
Well, I would think about drugs with a very narrow therapeutic window, drugs where it's difficult to measure the pharmacodynamic effect. In an anemia drug, you can measure the hemoglobin and feel pretty confident that you have an effect, but you often don't have that option. If treatment failure or toxicity is what you're likely to run into, then I think you would want to have more confidence that you had real PK data.
Kirkita, please.
Yeah, I just wanted to add to that, to what Susan said. I think the other issue is also it depends on the indication, right? So if drugs are being developed, for example, Jan provided the example of the SGLT2 inhibitors, right? If the indication is for diabetes, glycemic control, then there may be evidence that the drug doesn't work if your eGFR is below a certain value, right? On the other hand, the same drug can be developed for the treatment of CKD, for slowing the progression of CKD. And so in that case, you want the data from pediatric patients with the condition, with reduced kidney function, not only the PK data, but data to support,
efficacy and safety.
Well, let's suppose that, our ability to get data pre-approval is not going to change that much. And so what we can change is modeling and simulation, which we've had a lot of discussion on here, but also with post-marketing requirements, which we haven't discussed here. And, we have the ability, obviously, to put that in guidance. I'd like to ask Lynn, it looks like some of the post-marketing registries that we have, one of the biggest ones we have is for pregnancy. And so can you tell us a little bit about what our experience would be if, in fact, we started routinely asking for post-marketing requirements for registries related to these drugs and pediatric and renal impairment?
Yeah, did you feel that cold rush of air that just passed through when Gil said post-marketing requirement? Because I felt it. So there are a lot of ways we could consider that, and I think that one of the follow-ups from this meeting is I think we need to get together and think about. But I'll throw the question back. So the answer is there are many ways we could consider collecting that information: pre, post, registry. We could consider a lot of those options. But I think if we're going to pull the trigger on any of that, and I think that's what I'm hearing from the entire group, is I'm going to ask the panel, what's your priority, right? We're not going to do it for every drug, for every condition, for every situation, and every pediatric population. But if we're going to pull the trigger, where should we be aiming the gun first? Please, Ash.
I think just going back also on the question earlier, to me, it always depends on the need of the drug, which population you're going to use the drug in, and what's the, if you look at the real world evidence where it could be applied, that may be a starting point to decide where you would need more data or where you would need to collect the data in patients with, pediatric patients with renal impairment. And then you have the constraints when you design your trials, what would be possible in the clinical centers around the world, also in the U.S., what kind of patients would be available, and that defines the limits. But then, of course, you can if you can have the registries after post-marketing, that's where you can fill the gap. But I think that you have to start off always to see where you have the need for the drug in patients with renal.
So Ash, I'm hearing a lot of different sort of priorities. Let me try and focus, and maybe Guido can respond. So I'm hearing first we know where there's a need. So if we could clarify more specifically which populations is there a need, and then I also heard in there sort of the things that are feasible and easy maybe we should try first. So I heard that piece of it too, but please, Guido.
I think a guiding principle that I would follow is preventable harm and toxicity, right? So if a drug has a narrow therapeutic window and has a propensity for toxicity, then that should be a prioritization.
Brad, please.
Yeah, the other thing I just want to say is, you brought up, what's easy? And I can tell you in pediatric nephrology clinics and academic centers, none of this is easy. And we just have to recognize that. And we talked about that in the break with Ron and with Nick. There's so many factors that are influencing the success or failure of clinical trials in pediatric nephrology centers. But as somebody, I'm the chair of the clinical trials committee for Naprotix, and we never meet our target enrollments because of all the factors that influence that. So we have to recognize that. And I think Jan's last point, that academia and the pharmaceutical industry has to work together, that no question we have to do that. But we've got to find a practical way to get these studies done that we think need to get done. And we're not there yet.
So, Brad, I'm glad you brought that up, because I think hearing that the priority is, look, the ones that we think are, maybe going to cause harm if we dose it the wrong way in children, and really there's nothing that's easy, and we got to figure out a way. So I think the panel, it would be very helpful to hear from the panel. how do we create that path? What is necessary for these studies to be in any way feasible? Susan, please.
I think I heard from the modelers that we didn't need that many patients, we just needed the right patients. And they needed to be across a range of GFRs, and that's reassuring. Now, I didn't say it's going to be easy to find those patients and who are perhaps having the drugs prescribed by well-meaning but guessing clinicians. But if those data could be collected, if those samples could be analyzed, if the assays to measure those drug levels were available from the industries that develop them, then you might have a path forward.
Please, Jan.
Yeah, I fully agree. And the question is, do we need clinical trials at all, or do we need the data? Because we can also generate these data, especially if it's PK, in a kind of open label setting, where we would not necessarily include these patients in a study. The question is, when do we really need these data, and when should we start with the data collection? And therefore, I agree. It's not really about, is it a post-marketing requirement or not? It's really the question, do we want to generate, or do we have to generate the data, and when do we have to do this? And if this would be in a post-marketing setting, for me, it would be part of the clinical development plan that has to be defined up front. So we are actually prepared, and that we have to generate these data. And therefore, the idea came up to, whenever we have a clinical program ongoing, to leverage this network and this ongoing study and the sites to then also have a kind of ancillary, maybe open label arm, where we would try to include already patients with renal impairment. And in case we have sufficient data at the time of approval for the general pediatric population, it would be good. If we are not there, we could then still continue, but at the same time ensure that we could already submit for the general population. And having, I think, guidance would help, because then we could better really plan how to include it and how to build the strategy. And I also agree with what you said yesterday. We haven't really tried, right? So we do not even know. We know it's challenging, but we also haven't really tried a lot.
I'm going to ask another question that maybe is a little provocative. So clearly we heard that it needs to be a collaboration between industry and those centers that have the patients, right? Whether it is CKD clinic, whether it's, NICUs, whether it's, acute care settings. How do we get academic pediatric nephrologists to buy into the idea that these data are important? Because BI is coming, or because Novartis is coming, right? How do we, because that's what I'm hearing. I'm hearing it has to be that way. How do we get it that way without, I think what I think historically has been the reluctance to participate in data generation from industry?
I think that it's a very complex situation. It's a lack of training on clinical trials. It's a parent perspective that is important to consider, because they want always the best therapy and they don't want to get randomized to placebo, etc. And there's also a big center effect, because you do things the way that the center that we trained taught you, and deviation is challenging. Pediatric nephrology is unfortunately at the bottom of the bottom when it comes to clinical trials. But the academic centers have an interest. And one thing I would like to say to industry, that because of the small number of patients, etc., we're not a priority to be visited. And that culture could change, right? Because you need to have, in order to make progress, we have to have access to the assay for whatever drug we want to study, and that is usually proprietary to the company. So many drugs are not available to be measured in a clinical setting. And there has to be a communication avenue. If you call a company abroad and may not even be able to speak the Japanese or whatever might be the right appropriate language, and then to make that connection until it triggers down to somebody with whom you could actually collaborate and give them a frozen sample of a test or so. I would invite the companies to be more open, also with respect to older drugs, not just with new drugs, because I think we need to establish the safety for all the drugs that are widely used.
Please, Brad.
Well, as Guido said, this is very complex, of course, and it's not the pediatric nephrologist per se, it's all the resources they need. I mean, there's a shortage of research coordinators to actually help carry out the studies, and without a research coordinator, you cannot conduct these studies. And I hear that across the board in the pediatric nephrology centers. Susan and I were at a meeting yesterday about another clinical trial, and one of the things that got brought up was about the education of our community, the parents and our providers, in terms of the fact that all is not well with CKD management. You know, we look at cystic fibrosis and we look at oncology, and when you enroll those patients, it's a life and death kind of situation, and families readily enroll. Well, in CKD, you know, I think the nephrologists have generally sort of wanted to share with their patients, you're doing well. But they haven't highlighted the fact that the lifespan of these kids is compromised because they have CKD, and they're going to develop end-stage kidney disease based on our current therapies. And so it really is a life and death kind of situation, and we have to communicate that better in a way that's acceptable. We're sensitive to it, but I think that will help encourage the enrollment if we're going to do clinical trials, and help encourage enrollment of these families so that we can get the proper number of patients, that critical mass, so that we can address the drugs in an efficient and a timely manner.
Thanks.
Just to add to that, I think that we need more non-inferiority trials and not so many placebo-controlled trials so that it's more acceptable for the caregivers.
I know Saskia wants to make a comment. No, please. Oh, sorry, Deepa, and then Deepa.
Okay, so I'm an intensivist. I think we are on the bottom of clinical trials, but no, but just one thing that I want to mention. One of the things, a couple of things have been mentioned, but one of the other things we've been building in the U.S. and in Europe, pediatric clinical trial networks like ADEPT and Connect for Children, C4C. What we have learned, and one of the first services we're going to roll out in Europe is feasibility across Europe, one contract, and we can approach 300 sites. I'm responsible for the sites in the Netherlands, and what we have learned is that companies also have an issue to find the right sites and to find the right doctors. And when there's a CRO that they don't know and sends them an email, they're usually very complicated, these emails. They're like, hello, Dr. So-and-so. They sent it to the wrong doctor, usually. And not by intention, because they don't know which specific doctor is treating within that department that specific renal disease. And then they are like, well, I'm not interested, and they throw it out. So what we've been doing in these networks is sending these. If we approach these doctors, we know who to approach, and then we can also explain why the importance of this study is. And the other thing is that, and I don't know I think we all need to be responsible, is we have to show that quality of care gets better when patients or sites enroll kids or even adults, when sites contribute to clinical trials. When patients are in clinical trials, not only those patients, but the whole department, the whole hospital, their quality of care gets better. And I think that's something that we need to tell our physicians, but also tell patients and parents. And that's like a general message that I think doesn't always come across, and it's much better than 10 years ago when none of the physicians wanted to participate because, oh, no, it's too much burden for their patients. But it has gotten better. But that's a general message that we should get across, independent of pediatric nephrology. So those were my two comments. And I think industry needs to start thinking about using these networks more than just trying to do it all themselves. That may really help.
So while
from an
go ahead.
industry standpoint, just let me say amen.
Okay, thanks, Ron. And before Deepa says anything, I do want to say, it sounds like what we haven't quite necessarily established, though these clinical trial networks are promising, is what I hear on the academic center side, as Brad is mentioning, that there isn't a point of contact because there aren't these research coordinators that have been identified that would be the conduit for, disseminating the idea to everyone. So, as part of that, I think we have to think about what kind of support is necessary to keep those research coordinators employed at those academic sites, because they're the ones who are going to be funneling all the studies to these centers. Please, Deepa, please.
No, and I think we are raising some key points here in terms of barriers on both sides, right? And I think, Jan, you said something that I think maybe got glossed over a little bit that I think is critical, is that we have to start thinking about this in the beginning when we're creating clinical development plans. And I think that is a push to say, hey, we need to be proactive about that on the industry side and start that ball rolling early. Because if you don't think about it, it isn't going to happen. And whether that be doing it in a protocol, whether that be doing it in a post-marketing setting, either way, if it's not incorporated and thought of early, there's less chance of it happening, right? And then I think, we've said these words a lot throughout the session, these data. I think we really need to understand what these data mean. Because this group is here because we are advocates, because we want to be here. But I think from a practical standpoint, what is essential? What's the data? How do we write that into protocols so it gets implemented? Then from a site perspective, is it clear? Are we making these things clear at investigator meetings? Are we having investigator meetings? Are we making sure the points of contact are there? I think so many times we use and rely on our clinical trial centers. I have. They do my contracting. They'll get the IRB ready. They'll do all this. And is the research coordinator really engaged? Is the PI engaged? And so I think the onus does fall, but I think looking at this almost like a playbook and saying, is step A to B to C actually getting connected? Because I think what you're identifying and what I'm hearing the panel say is there's disconnects in these steps. Just my 2 cents there.
Ash, please.
And just to add to what Deepa was saying, I think that the data is very important. What you get out of the centers, how will you use them? Because unless you have all the right information, at least from PK perspective, you would not be able to use them in a model anywhere unless you have all the right information around it. Time of administration, time of sampling. There are a lot of things that are needed to be able to make this successful.
Lily, please.
Sure. Maybe to add to what Saskia was bringing up, as well as Deepa, is there a role for platform or umbrella trials here? I think we're talking about open label PK safety studies. So in order to make this efficient in terms of protocol development, IRB review, to help the academic sites, I'm just wondering if is there a role for collaboration between various industry sponsors to allow for single protocols to collect these type of data. And as new compounds come, it can be added to the protocol. I'm just wondering if anyone would comment on that.
That's the big question. and for sure, I think whenever we would take PK samples and a patient would be on different medications, for sure it would make sense to assess multiple and to do PK sampling. I think, of course, there would be support. Of course, the question is who would start this initiative and who would kick it off. I think this is the difficult part. But also just to provide one comment to your previous question, I think one of our learnings within Boehringer Ingelheim is, and this also goes back to what you said, we really have to start having these discussions very early on and also bring in experts very early on. So we cannot design studies without aligning with experts and then assume that this is feasible and doable. And therefore also based on the collaboration with the FDA, I think what we really appreciate is for some pediatric requirements, they are phrased in a relative open way. And this allows us to really go back to the experts, discuss, and come back with a proposal instead of having very specific questions, and then we have to try to build a protocol around. And therefore, this is what we really appreciate, that we feel that the FDA helps to let us also know, let us speak with the expert, and then come back and have a discussion for specific medication.
Brad, please.
Yeah, I'm going to follow up on that. You know, I've been involved in clinical trials for a long time, and the ones that work best, that take everybody's issues into consideration, are when the nephrologists get involved very early in the development, as opposed to getting an adult protocol. Let's fix it for the kids. That doesn't work. So I'm absolutely in agreement with you. The earlier we can get involved, the better.
Susan, please.
I want to say amen to what Brad just said, but I also want to go back to, when do we need a clinical trial, and when do you need sufficient data to understand dosing in children?
Yes, that's a really important point. And so I think if I can try and clarify a little bit for folks. So in the context of, you know, what I would describe as a trial is just a means to collect data in which we have to get consent, right? I mean, so it's not necessary when we say trial, you know, or PK trial, it doesn't necessarily mean that it has to be, you know, controlled, and we have to, you know, collect secondary or primary endpoints, and there has to be a long safety follow-up. So a PK trial could be something, as Lily described, as, you know, a platform trial. Just submit those patients who you have, you know, collected data, and you have to get consent to send their blood. So it's a trial in that sense. And it's, you know, collaboratively designed, developed, implemented. Or it could be, you know, 6 patients for an individual company, because we think we need 6 patients less than 2 years of age to confirm the model that we've created. And then you're kind of done there too. So that, when I mean trial, it's in the broadest sense of trial. It's really a way to collect clinical PK information in this particular situation. Let's see. Sonia.
So I completely agree with you, because I have training in PKPD, but I think that most clinicians have very little training in PKPD, and what we have been taught in medical school is the randomized control trial is at top of the pyramid, and we need a randomized control trial in order to change interventions. And, you know, again, in my anti-infective world, the reason that precision dosing hasn't become commonplace is people are saying, where's the randomized control trial that says that this improves outcomes? And what I have noticed is, and this goes very similar to what everybody has said before, is that where we've made the most progress, at least at my institution, with clinical pharmacology studies, whether it's PK, PD, and pharmacogenics, is that we have to have a clinical champion who has either seen something bad happen to their patient because there was either underexposure or overexposure, or a patient or the patient's family, where they, you know, they're getting drug levels on their biologic, and they're like, my child is not getting better from their disease process, and they go to the clinical pharmacology division and say, we need to do something about it. So I think clinical champions, patient champions, and I've been pleasantly surprised that once we approach a patient for a clinical study, when you explain that there are no data to drive the drug dosing that we are giving them, we get 70 to 80% rate of enrollment in these studies. So I think when you explain it to patients that we're doing our best in empiric drug dosing, but we have no data, I think we can get whatever the data that we need to get to do these studies, and that we need to reframe for physicians that randomized control trials may not necessarily be necessary to make changes in our interventions.
Thank you. Guido, please, and then Susan.
I wonder whether there's also a role of the colleges and the licensing, because ultimately we need to change behavior of the treating physicians. In Ontario, in Canada, we now are required to have quality improvement projects, and we have to actually document that we are doing those. And sometimes the barriers of the ethics submission and everything are so big that people are giving up. But if you have a college requirement for conducting quality improvement projects and improving drug dosing and improving the safety and efficacy of drugs in renal impairment is a quality improvement. So maybe we should even label this a little bit differently and call it quality improvement to improve the safety of the drugs rather than always talking about a trial.
I want to play the opposite side. We're still talking about a very rare disease. You're talking about renal insufficiency in children, and we might wish for a very broad-based, every clinician in the country has to participate, but in reality, there's going to be a network of centers that see these patients and are prepared to do it. And so you have to have your eyes open to that. Who sees the patients?
Brad, please.
And I think along the same lines, there's a limited number of patients and more protocols that are out there, and almost not enough patients to fill all the protocols. So that's another challenge that we're always dealing with.
So that brings up a big issue for industry, and since I didn't have a slide, these are my opinions, not the opinions of my company. Platform trials are, in theory, something welcoming, but when we have a requirement to do evaluations in children, how do we know where our asset will get prioritized and will it fulfill our obligation? And the struggle with too many options for small patients, that whether it's kids with CKD or oncology patients who might develop renal insufficiency, is a big challenge for us. And I fight being nihilistic about that challenge.
And Adam is a pediatric oncologist, so I would, maybe take a playbook from what the oncology COG leaders are now thinking about, and that is that, being enrolled in a clinical trial doesn't necessarily exclude you from participating in another clinical trial if that's not going to affect the outcome of the other trial. So we've seen automatic exclusions, like you're already enrolled in a clinical trial, you're out. But for purposes of collecting a PK sample, why in the world would you exclude, that patient? Again, unless there was some reason that it would interfere with the, dosing or whatever. But I think that's another area that when there are, smaller populations that we're interested in studying for multiple therapeutic therapies, that can be also a way to, at least in the setting of, this discussion, which is we just need some PK on renal impairment, Yeah,
I think that's a great thing to highlight. And you're right, for children's oncology studies, there are multiple studies that can overlap, and you can be involved on your main therapeutic trial, but then on a research question, separate trial at the same time. You just triggered a thought, which is, one could imagine that if you're enrolled on a therapeutic trial, instead of being tossed off of the treatment arm because you develop renal insufficiency, one could imagine that there's a rescue that, yes, you developed renal insufficiency. Now, even though PK might not have been involved in that therapeutic study, now it will be, and there might be a pathway for you to continue on that therapeutic trial. And I'm not sure we really thought about that yet, which is what you're mentioning. And then we would get the data, and even if it's a limited number of patients, have some idea what to do for these patients later on. I know we're all so passionate. It's a terrible, and we all know it's a terrible situation when you're trying to literally give life-saving treatments to someone who develops a problem, and you're just going like this and saying, I have to give a dose, and I don't know. Thanks.
Gil, please.
So circling back, I guess now that we mentioned that we have a very small number of patients, circling back to the modeling and simulation, I hope we can do better than the 15%. 15%, that was 15% of drugs that really should have had renal recommendations and labels. Only 15% had them. 100%, we're not going to get there, but I hope we can do better. Certainly the modeling is sophisticated enough. There was discussions here about ontogeny of transporters. all those things have actually been built into some of the PBPK models we have now. The FDA is accepting modeling of drug absorption for bioequivalence. If you look at what's going on in the Office of Generic Drugs, and we hope to certainly apply that to drug absorption in pediatric patients. So complexity is not an issue. And so the question is, can we use modeling and simulation to? I mean, there are some situations in which we'll be prevented from making because we have such a small number of patients, or we can't really, we don't feel comfortable because from a safety perspective, a risk-benefit perspective, that we can do that. But I hope that we can start applying that more and be able to actually provide dosing recommendations for clinicians in the majority of drugs that need them in pediatric patients.
Nick?
Two completely unrelated questions, and I'm not sure the FDA would have a solution to either. But I really like these comments about having a clinical champion, and also the idea of the licensing bodies raising the importance of this work. Thought needs to be given as to how the sort of academic importance of this type of work can be increased. So Brad's clinical coordinator, who's going to be absolutely key to this work, decides that he or she is going to spend their time on this project and not some new exciting gene therapy for a podocyte gene mutation or something. So it's how the academic importance of that can be raised. That's my first question. And the second is just a thought. In every company in the world, or in their laboratories, there are freezers full of samples, duplicate samples from multiple studies, which will involve lots of children with CKD. And I just wonder if there's a way we can think about utilizing those somehow. Difficult, but you'll have very accurately collected dosing, concomitant meds in a DDI. There's potential there, maybe.
Yeah, thank you, Nick. Curious what our panel thinks about increasing the presumed, value to the individual investigator for collecting this kind of information. We heard from Guido that, make it MOC, right? There's silly QI projects that we have to do. This would be a real QI project, right? We'd really be able to affect a change in labeling and drug dosing in children with renal impairment. Adam, please.
I want to say that no MOC project is silly.
Sorry. Strike that from the record. I did not say that.
As someone who was in academia for 2 decades and recently went to industry, I'd actually like to answer the academia issue because I think I have no secondary gain other than how important it is. I talk with a lot of colleagues, and I think this is a type of forum where we have to start acknowledging what might be a crisis in support or lack of support for academic work in pediatrics. This is certainly incredibly important. It takes 6 to 9 months to get a cancer protocol through many academic IRBs. Also very important. I'm not going to say we should rate one versus the other, but the dearth of supportive resources in academic pediatric medicine right now is at a level that I think many people on the panel will say is, you could describe it as a crisis. How to get better recognition for that across the board? I'm not sure, but I'm certainly happy to publicly state that it's time that on a national level we talk about it more.
Again, to follow up on that, yesterday the match came out in pediatric nephrology, and 46.6% of the spots went unfilled. So we have a shrinking pediatric nephrologist population. Everybody's focused on their RVUs because the bean counter said you have to generate these amount of RVUs, and so it's a crisis. It is a crisis when you look at academic productivity, and which goes along with these trials.
So one way to answer the question without just throwing our hands up is to incentivize academic centers to support these rare populations the way they deserve, much as I think health authorities have incentivized industry to pay more attention to pediatric drug development. And, that might be multiple different seminars and discussions on how to best do that. But I do think we're at that inflection point where we can't just rely on the heartfelt passion for our young colleagues to go into pediatric subspecialties and do this type of work, and we need to have a more incentive-driven approach.
Please, Gil.
If we're ready to move to a different topic, I wanted to address Saskia's earlier comment about her computer program. And so I found that very interesting, and actually the FDA has a lot of experience with co-development programs. Now, co-development is usually a drug and a test, but in fact, we have drug development tools. We have a qualification process for drug development tools, and we have other pre-approval compliance programs with basically scientific computing. And so I would encourage sponsors to think broadly about how they could maybe apply those types of computer programs and modeling that would continue to collect data and become perhaps more precise over time, and to think outside the box, because the FDA is open to working with those types of situations.
Okay, I have another question that will require each of our panelists to respond. And I don't care if we start with Kirtida and go that way, or start with Guido and go this way. But, I mean, these last 2 days, and for Brad and Susan, even just the morning has been really, I think, illuminating in a lot of some of the challenges that we have identified, and I think created some potential strategies to address those challenges. So if I could ask each of the panel members to tell us, what of the things that you have heard and considered yesterday and today you think is the most important thing we should act on following this meeting? And it's probably not fair but I'm gonna pick on my FDA colleague to start.
It's a tough question. I think one of the things for me in terms of the discussions, which have been amazing, in terms of what do we do for dosing children with renal impairment for drug labeling, right? One of the things I think I wanted to go back to is this question about, the degree to which we can extrapolate data and leverage adult data given all the challenges that people have described for getting data in pediatric patients. And to me, one of the kind of low-hanging fruit, which I feel we discussed to some extent yesterday, but maybe need to discuss a little bit more and get more input from people on, is this issue of, proportionally dose adjusting for reduced GFR when you have data in adults and data in kids with normal kidney function. So, for example, if you have data for drug clearance as well as efficacy and safety for adults with both normal kidney function as well as various degrees of kidney, reduced kidney function, and you have a 5-year-old with the same condition receiving the same drug, but with normal kidney function, and you have PK data from the study, from a study for that 5-year-old, can you then, translate the the data from adults for, if a drug is cleared and your clearance is 50 in an adult and it's 50, in a child, can you then translate that data into pediatrics? I think that, for me, was like, a key question that I feel we may not have fully addressed, but that maybe could provide, some information on how we could move forward with what data we currently collect.
Right. So what I'm hearing is the situations in which actually no additional data in children are needed. Like, can we put some better guardrails around or construct a clearer approach to that? And I think we did talk about that yesterday. I'll remind folks of that in the summary, but, is there a paper or a guidance that's, necessary to make sure people understand that and to, clarify only those situations where we think we have high enough confidence?
Yeah, and also, do those, does it have to be only in, products that are close to 100% renally cleared? If you have the data for renal impairment for even drugs that may be less renally cleared, but, have data for adults and some data in pediatric patients with, normal kidney function.
So Kirtita's describing the triangulation, which I think we've all indirectly discussed, but let me just clarify. So the triangulation is you have adult normal PK and normal renal function. You have pediatric data in the drug with normal kidney function, and you have adult renal impairment data. How much pediatric renal impairment data you need? Which situations are we comfortable in which none is necessary? And that deserves, I think, what Kirtita is saying, that's a priority that deserves further discussion. Okay, Gil.
So the statement that stuck with me from day one was that on the panel, in panel number one, was Dr. Tuckman saying that the recommendation that we would make today may not be the recommendation that we would make in the future. And so the responses to that could either be to throw your hands up and say, oh, well, the science is changing, and we're understanding more, so why do anything? But I think the opposite to that is to say, well, okay, well, let's make a recommendation today. Let's help clinicians make a recommendation today. If we need to change it in the future, we can, but in fact, let's make a recommendation.
As one who's been a pediatric nephrologist for more than 40 years, I first want to say it's terrific that we're actually here, that we're talking about kids with renal impairment and how to provide them with medications in a safe and effective manner. So that's terrific. That's an accomplishment. I agree with what Kirtita said, the triangular thing, and modeling. I mean, I think I've heard more about modeling, and I think that is a way to address the fact that we're never going to have a lot of data from children with renal impairment. And if we can take the knowledge we have from modeling and the few samples that we will have, I think that would be great.
Thanks so much for the question and for this whole panel and this whole couple of days. There's been so much that I could mention. I'm just going to highlight two things. One was, I think, from this morning when a colleague said we need to really check ourselves with how precise we need to be. And something we talk about in awards about like clinical significance versus statistical significance. And sometimes I worry that we're getting into things so small that we disappear. That's a poem by one of my profs, and Dr. Ammons. So that's one issue, is like how precise do we really need to be to just be good enough, which is not to be complacent, but to be pragmatic. And the second was a comment yesterday about the inclusion of adolescent patients as they go through development, whether it's from young adolescents to older adolescents or adolescent to young adulthood. I think there's a very big topic of mine that many in this room have met to discuss about, and having clarity on how to best navigate those waters with regard to renal impairment would be very helpful. Thank you again.
I thought the workshop was really interesting. Learned a lot of things, especially about this very few labels having information for children with renal impairment. Working on the IQ side, what we have been talking about a lot is that, if you build a PK model early enough, I think some of the colleagues have done it in the industry, and we're trying to do it also in our company. If you can build a PK model early enough and can make a prediction on what would happen with different degrees of renal impairment in adults, and then you basically test it out in the drug development in the adult side. And if you have a good PK model which gets informed as you go along for adults, then you make predictions for children. What should exposure look like in a child with normal renal function? And if you're addressing that, eventually to go towards the, make any assumptions on what would happen for a child with renal impairment. So for me, any data we can get, which in patients with renal impairment, is only confirmation of what a model should tell us in our drug development. So we make a model, and even if it's scarce data, it only helps to give us more confidence on what we should be expecting, and that should help to have a better label in the long term.
Thank you, Ash.
Yeah, I agree. It was a great and important workshop, and shows that there's a clear need that we have to do better in the future. And this means whenever it comes to pediatric labeling, there should be information for pediatric patients with renal impairment. And I think we would really need the guidance, and I agree, where we could do modeling only if we have adult PK data with normal renal function and impaired renal function, and PK data in pediatric patients with normal renal function, and then extrapolate to pediatric renal impaired. So I think this would be important to define when we can do this, but also when would we need additional data. And having clarity, I think, on this would, from my perspective, help in the future.
I agree with everything my co-panelists have said. It's been an excellent even half day for me. And you talked about using modeling to enhance your confidence, or to move into a space where you feel you can't measure. But I think if I pushed you a little harder, can you create a program or a system where there is sparse, opportunistic sampling of children in this rare population, so that you actually can validate these models? So either you fix them if they're not quite right, Gil, or you have more confidence, Jan and Ash. So you said we did it right, and we're done. And we did it in 10, 20 kids, and that was what we had to do. The underfunded Best Pharmaceuticals in Children Act Pediatric Trials Network is not really set up to do that, because they're looking at normal kids and more common drugs and older drugs, but that might be the place where you create an appendage that makes that possible.
Thank you, Susan.
I think I would like to comment on a slide that Saskia was showing at the end of her lectures, and say no child should be denied drugs that are applicable unless there's an absolute contraindication. I obtained a PhD in clinical pharmacology because I wanted to improve the efficacy and safety of drugs and that I'm using as a pediatric nephrologist. And I think that access to assays is a very key enabler, that we should have a broader, and I shouldn't be having to receive the message that we stop measuring this because the clinical trial was finished. I would also like to just point one more point out, and that is we cannot just look at PK data. We have to also look at PD data, because you may have disease-specific therapeutic windows. So, for instance, mycophenolate mofetil, so the MPA levels for a transplant patient have to be, or the AUC has to be between 30 and 45 milligrams times hour per liter. But for a nephrotic syndrome, they have to be higher, right? And I think it's equally important that we also continue to evaluate the efficacy of the drugs, and some diseases may require higher than adult AUCs.
Please.
I fully agree with what you have said here. I think this is why I talked about data earlier, because data means you need a big breadth of information for it to make any sense, to be able to use it in any meaningful way. Because whether it's a disease or whether it's what dose was taken, when it was taken, all that is very important to make any value out of it. Otherwise, I think how my modeling colleagues say, excuse the pun here, shit in, shit out.
Guido just said a dangerous word also. He said nephrotic syndrome. And I think I'm just going to let it hang there, but your PK and your PD are going to be
very different.
Very different.
Yep. Thank you, panelists. Now, I'm going to do something that's a little off script, if that's okay, Bakri. So, we have a lot of panelists who participated yesterday who aren't up here on this particular panel. And it would be very useful if those of you who are sitting in the audience but participated in any of the panels, if you had in your head a priority that wasn't articulated by one of the folks up here, we would love to hear your thoughts as well. Shamir, please.
I'm going to make kind of a very broad statement, but I'm going to say it anyway. I think as we're writing protocols, or if we have protocols, anywhere you see exclusion for renal impairment, you just remove it. And instead, the default should be that, and now we have to think about why we should put it back in. Why would a patient with renal impairment be excluded for safety or lack of efficacy or another scientific or ethical reason to exclude them? But the default, as we're writing them, should be pediatric patients with renal impairment are in. And now we have to think of a reason why they shouldn't be in. That's not related to necessarily feasibility or, pragmatic issues with conducting the trial, but really based on their safety and ethics of not having them in the population.
Thank you, Shamir. Excellent. I see Jiping in the back.
It's not a summary of what I heard, but it's really a reflection and commitment I made to myself as an industry participant of this workshop. The panelists, the clinicians particularly keep on talking about the low in the list, and the industry people express the pain of finding them. And the network, ADEPT, C4C, as a platform to help with that is all good. But what I have learned when I transitioned from Clin Pharm department to a dedicated pediatric drug developer is that when I was in the Clin Pharm, when I designed a study and demonstrated instead of 20 kids, we can have 14 and get just as good data, I was very, very proud. But what I learned when I get into the role of pediatric expert and review what we have done is that I was just shocked to see that what influenced the speed and the cost and somewhat equivalent to the success of the trial is in the trial operation for pediatric subjects. It's not necessarily the number of pediatric subjects, but where you look for those patients, how you network, how you set up the trial, the recruitment, the on-site initiation costs, etc. Those actually have even bigger impact in terms of timeline and the budget and / success rate of the trial. So when I heard the commitment from clinician, from academia side, I think as an industry participant, I made this obligation, commitment to myself is that in industry, we often separate the science and the operation side. But the operation can be highly science as well. So I will work closer with our operational side to reach out to the people who care about this population, care about overcoming this challenge together to do a better trial. So that's just my self-reflection, thinking about what I'm going to do beyond the science and learning that we talked about here.
Yeah, wonderful. I mean, I think that's one of the things that, I guess, is an important function of a meeting like this, is that we help to connect dots where maybe they haven't been connected before. And so hopefully that has, you know, in each of our roles considered where we could be better connecting those dots. So thank you, Jiping, for that. Let's see, who else did I see? Oh, I see Deepa and then Hal. Yeah.
So, you know, you asked what's the priority moving forward, and I think this workshop's been tremendous in really starting to peel the onion. I think we're going through layers, and we're finding barriers, we're finding opportunities. I suppose the priority is not to lose momentum. It is really to get together, get next steps, and rather than continuing the dialogue on principle, to put it into action. And I think that's what resonates with me.
Excellent. Hal, please.
Yeah, I really appreciate all this wonderful discussions we had yesterday and today. So, one situation I think I wanted to emphasize that, I agree with everybody saying that we're really dealing to most pediatric patients with the renal impairment. They are pretty rare conditions. It's always challenging to enroll them into a trial, even though we understand the necessity. So I'm just trying to advocate the possibility of, getting something done through modeling simulation as the first step, and then putting this out to the community. Of course, we need to select the patients and select the diseases, select the drugs with, let's say, flat ER relationship, so there will be very low risk scenario to start with. And then we are collecting data after we started using them as a feedback loop in the future to fine-tune our model and then potentially change our dosage recommendation. I'm just going back to what Gil said. Maybe we have to change that dosage recommendation in the long run, but we have to have something to begin with as a starting point. I think that could be something that could be a practical solution. Where we're facing challenges in the trial design and getting patients into the study, and in the meantime, we do need some information to guide the usage of this population.
Thank you, Hal. So learn and confirm is what I've learned from my modelers. I know George has a comment too.
Thank you. Yeah, I was actually thrilled to kind of get to meet many of you and work with you over the last 6 months. And we put a lot of time into setting up much of this study. But I think there's probably 350 pediatric nephrologists that don't know what you guys do. And I would suggest that your summary, Lynn, that you're going to make for us, we can go through it maybe with a panel, and then I think we should disseminate it through the ASPN to all of our nephrologists so that we can all be on the same page and be available for industry to get some of these medications out to the docs who really take care of the patients.
Terrific. Excellent comment, George. Does anybody want to go? Saskia, please.
Yeah, well, thank you again. This was an exciting workshop. So my take-home message is that there is literature out there, and I think we should indeed start to pull out these data, really dig a little bit deeper than superficially looking at the abstracts of these papers and see if we can actually figure out if there is a age point at which extrapolation is not possible anymore, or if we can extrapolate from adults to kids, and to also figure then out where the information gaps are. And the second one is use these data and existing data to further show that PBPK modeling can be used, so that it can be a tool that the regulator can actually demand from pharma when you submit your application. But the first thing I think is I'll go and talk to my PhD student and see if we can get a next student to start looking at those topo PK data.
You heard it. Saskia's volunteering a PhD student. Maybe. Okay. Any other? So thank you to the entire panel. Really insightful and I think excellent discussion. Thank you again. let's see, I think it's the first step, right? It's the first step. All right. So we'll give a second or two for folks to are you moving that or am I? Oh, perfect. Okay, perfect. Yeah, so I have I don't know if it's enviable or unenviable task to summarize the proceedings of the last day and a half and to provide, I would say, this is, even less this is even more of a disclaimer than I usually have, which is that they represent my views and not the FDA's. Here, I'm not even sure if they represent the room's views, but I'm going to present them anyway. So let's see if I can move forward. Yeah. So Shamir put this slide up at the beginning, and the idea was he put up 3 bullets for us that says, here are what we hope to achieve in the next day and a half, to develop a shared understanding of the key challenges and knowledge gaps limiting the establishment of accurate dosing for drug products in pediatric populations with renal impairment, generate potential strategies to overcome these challenges, and then articulate future directions for a shared commitment from stakeholders. And then he provided a slide that included, the challenges that we were all sitting here recognizing and acknowledging at the beginning of the day, the beginning of yesterday, I guess. And then, I want to also remind the data that were presented, our fellow Sabrina, who did the work, and Saskia's fellow who did the work, to say that the current state that we're in, which is, do we have dosing recommendations and FDA-approved labeling for pediatric patients with renal impairment? The answer is clearly no, right? If you, depending on the, data, you say 10%, 15%, it's really, not where it should be. So, in terms of a recap of session 1, what constitutes renal impairment in pediatric patients for the purposes of PK characterization and drug dosing? I think we answered this question about did we answer the question about what constitutes renal impairment? Now, I threw this up there because it's loosely based on what, we know the U25 equation tells us, is that we have confidence less than 75. Now, I'm not here to debate even at this point whether it's 75 or not, but we know that we're talking about some level of renal impairment, and it's not those patients who have normal GFR or are hyperfiltering. And then what estimating equation could be used? So for renal impairment, I think we came to a consensus that the U25 down to birth with appropriate measurement of creatinine and use of the appropriate K value could serve as a really good estimate for GFR. Of course, we have to use IDMS standard creatinine, and it's not appropriate under certain situations, and we need to be clear about what those situations are. We need to stick with the same estimating equation once we started it in a clinical trial or in a PK study, that CKD epi is not appropriate, and the bedside Schwartz is probably outdated. I think those are things that came out. And then if we're talking about measurements at baseline in a clinical trial, I think we also heard collect a measured GFR. So do the iohexol, collect the cystatin C and creatinine to calculate your GFR. That really gives you high confidence in what you're starting with at baseline. And then perhaps follow up only with creatinines and then calculate your eGFR using U25. There needs to be a plan to educate the community that U25 should be used, even in adolescence when the adult estimating equation is different. I think that's what I heard also. But again, with the caveat that don't change things midstream, right? But start educating your clinical trial groups that this is the equation that should be used. And then I think there are some research questions that we heard also, like, where are the places where U25 maybe isn't the greatest? And we heard, certainly in places where we think there's hyperfiltration or there's, normal GFR. So maybe we need refinement of that, or in fact to consider using other estimating equations. This notion of gaining information from worldwide review of data is a theme that I think you'll see in basically every session. So then in session 2, we talked about how do you translate from adults to pediatric patients with renal impairment. So what circumstances can we be comfortable in leveraging, right? And I think what we heard as a wish from some of our panelists is, could we go beyond, what we described as, where our comfort level is? So, the dose adjustments based on proportionality assumptions from adult to pediatric without any additional data. So first and foremost, what we heard was there has to be confidence that the diseases and the response to treatments are similar, right? That's kind of the first step. Then in terms of drug considerations, I think we also heard, look, if it's fully renally cleared without extensive metabolism, then we feel comfortable. Are the pharmacokinetics, and I put in quotes simple, and I only put linear as an example because I'm not sure that that's the only situation in which the pharmacokinetics could be described as simple, but I think as well understood or characterized is probably the better description. There are, limiting assumptions without data, to greater than age 2, I think is what we hear, where maturation is less well understood, less than 2, and that the impact of renal clearance, and differences in physiology, for example, total body water, may impact our ability to have confidence in the adult data. And then conditions where the dosing of the drug might need to be tightly controlled. And that's not just for safety, but also if you have, a problem with efficacy if that dose is dropped too low. So I think these are some general considerations that came out of session 2. Let's see. Okay. So what about those patients less than 2 years of age? So what I'm hearing is there's unlikely at this time to identify a model that will fully provide an estimate right now for children less than 2. So that's a place where we need to do some work. And I think there was a question about whether or not we could leverage data in patients with AKI, for example, neonates with AKI, to sort of fill in that under 2 population. But, I think the last bullet, concomitant medications, inflammation, total fluid delivery, CKD on top of AKI, those may really affect our ability to accurately measure eGFR in the pediatric AKI population, and maybe that's not the best way. But it was brought up, and I still think it's an interesting proposition. And then really, I think before we even consider whether or not we could use an AKI population, we also have to understand the limitations of the U25, which is, again, that wasn't really validated in patients with AKI, it was CKD. So again, some real hurdles to overcome if we were going to consider a different population to help us develop a model under 2 years of age. In session 3, was that yesterday or today? That was today. Okay, right. So this has not been reviewed by all of you. I was just taking notes this morning. So what I felt like I heard, and I'm presenting to you now, is that there was pretty good consensus across the room that model-informed drug development, that the use of modeling and simulation can be used to support identifying doses and renal impairment in children. And in fact, I think it's more than can be used. I think what we're hearing from many in the group is that it probably should be used. And that, one thing that came out, and that maybe not so much in the panel, but, if we're going to gain understanding and comfort in how these models were created, what data went into the models, and how these models will be used, whether they're fit for purpose, I think that requires that we start sharing that information. So if you're developing models in your organization, that you should be sharing those with FDA or other regulatory authorities so we can take a look and, critically review those models. And I think just having more heads reviewing those models will help, I think, speed up the acceptability of these models. there really needs to be data to increase the understanding of the mechanisms. That's those mechanistic models that, that affect dosing in pediatric patients with renal impairment. And the data that I heard doesn't have to be just in the drug, in children and adults to fit the model, but we should be leveraging the data from what you may already have. Drugs in a similar class with the same mechanism, maybe approved in children, maybe you have some PK data there. So don't limit the idea that data would have to come from only adults who are using the drug in the adult program. And I have to say that I think if I was taking out the main points that I heard, you know, from this morning, there were two major areas of data collection need. The first is to improve the models with the data that are already available. And I think that, that there is a need to figure out how we can collaboratively come together to review the data that are already available and figure out how this can be conducted, what funding should be used, and also to develop a priority list for which models and which drugs in pediatric patients with renal impairment should be sort of started on first. And then the second area of, I think, major data collection need is data to confirm the models and those predictions for specific drugs. So these requirements may vary and may depend on the confidence in the model and may depend on the actual drug that is under investigation. And we also heard a potential approach there that it should probably be a model-based approach as well, but we're probably going to need to have data collected. And I put post-marketing, it could be pre-marketing too, but to confirm that model and then to refine that model further for subsequent programs. And that this could be done through, potentially through requirements or requests to industry, not from industry. And I think that the major research need here is then that we really need to, if we're gonna be serious about creating these models, then we have to review the data that are already out there waiting for us to be reviewed, to put those into the models to see whether or not there really is a need for additional data collection. And then I just put a placeholder here because I didn't have time to write while we were chatting, but I think what I heard from the collected group is that, number one, there is real momentum to move and advance this area forward. And all of the suggestions for priorities from our panelists in the room we'll put out later. But I think what I heard was that there isn't anybody in the room who said, forget it, this is impossible. I think I heard that it might be hard, but I also heard that there's commitment from those in this room. And I would say, not just from academia, not just from industry, not just from FDA, that there was, I think, broad support that we have, I think, some work to do, but that work is so important to treating these children, this special population of children who have the right to access medicines that are actually safe and effective for their level of renal impairment. So summary. All right, so I put this all on one slide. So the first bullet that Shamir had in the day yesterday was to develop a shared understanding. I think and I'm going to say for me, I have a much better understanding of the appropriate estimating equations for eGFR, and that's like fundamental to how we're going to assess renal impairment. I have a much better understanding of the utility of MIDD in this whole process, and I also have a much better understanding of the data gaps and the conditions where confidence is higher. If we're talking about the second bullet, generating potential strategies, so we've talked about developing approaches for data collection in individual drugs and a platform or plan for collective review of globally available data to improve the accuracy of the available models, and again, to have confidence in the whole idea that a PBPK model can be used. And then finally, to articulate future directions for a shared commitment. I think first, I'm going to invite informally and maybe formally after this meeting to convene our planning committee to consider a manuscript to publish the learnings from this meeting, and as George very nicely put, so to educate the rest of the members of our pediatric nephrology community. And then to move forward with some of the strategies that we've already identified and the priorities that were described by the panel and folks in the room. Before we adjourn today, I do want to give special thanks. I think as Brad put, and George put, it was a lot of work to get everybody together over the last 6 to 8 months to get us all on the same page, to go to those planning meetings that FDA was scheduling right in the middle of your clinics, right in the middle of your, meetings with your clinical trial teams. So thank you for all of the speakers and panelists, and certainly for all the attendees in the room and out there in the virtual world. I know that we had over 500 people signed up, so thank you for attending. I have to give special thanks, though, to our FDA and University of Maryland CERSI staff, Denise Johnson-Lyles. If you wouldn't mind standing up when I call your name, it would be great. So Denise Johnson-Lyles, Denise, she was herding the cats for the last 9 months. And as well, Erica Pine, who was also helping. Thank you, Erica. On site over the last couple of days, Jacqueline Yancey from DPMH. Thank you, Jackie. And also Michonne Payne. Oh, okay. Hi, Michonne. Thank you. Thank you, Michonne. Thank you. We have folks from Dana Hamill from UMD CERSI and James Pauly from UMD CERSI. They're not in the room, but thanks very much to their support. And then, of course, Julie and the rest of the OND meeting support team, they were here to help. This is one of the first on-site. Yeah, Julie, please. Yes, please. Thank you so much. We have just started coming back to gather in person, so without the OND meeting support team, we wouldn't have been able to convene the meeting here in White Oak today. And with that, I guess I get to officially adjourn the meeting, and thank you all for participating.